Alina, nice meeting you. Thanks for coming, guys. It's great to have you here, and let's go to fire up the audience. Okay, so thank you. So I'm going to ask both of you, and then so I think that it's going to be cool in the afternoon. So I guess you guys have played more or less on time. Yeah, I mean, maybe what I'll do, I'll just stand up, when it's 5 minutes to the end. But then you know that it's not to the end, it's still 5 minutes, right? So it will be more civilized way. Yeah, exactly. And I'm going to stay on. So we are going live. So I'm going to say that if you ask questions, you can send me an email. And then if there are questions on the email, I'm going to read them out.
Good idea. Yeah, I think questions mostly will be on the first part, where you talk about
Yeah, depends on what the audience wants to ask. It's nice, right? Yeah, you can do it. I don't know, I see you so much everywhere, that I feel like you must have been at our home, like.
now or never.
You did. Okay, that's good. She's actually fantastic in taking. Yeah. Okay. Good. Thank you.
Yes. I'm also German.
I'm not German. No, I'm just Polish. I'm Croatian.
Almost 40 years. Yes.
Okay, so guys, also, would you like?
I don't actually know anything about the system. It cannot be very.
Ah, voilà. Yeah, okay,
yeah,
okay.
we have everything.
stuff is a little bit so bad.
When it's
Yeah,
green,
so it
then
goes
you are-
green.
Nicolina knows. But just
Yeah, I guess.
plug in
Please tell me.
play, maybe.
We probably just try to.
Oh, you have this little one. Isn't this kind of,
Yeah.
well, hard to see?
Okay, let's see. Let's go to the beginning. Okay. Is enough, you think?
Let's see. Seems to be on,
Let's see.
but there we go. Yes,
Let's see what it does if I do this.
there we go. My God.
Wow.
Okay, cool.
It's like this. This stuff is so weird.
I know, I heard them
Ah,
already.
you can also look
And
there.
you can look
Do you
there.
have a pointer?
Yes, we do.
I mean,
Pointer
Here is
is
a pointer,
here.
yeah.
and here it's. What is that? The switch?
You need this thing onto your machine or not?
Do you
Hello,
want
hello.
this-
Yes, it works.
Do you want this in so that you can use the...
Yeah, why not? I like
Yeah,
to
it's,
try.
good.
Never tried it.
You never tried?
Never tried
It's
this
very
with
convenient.
that you
Yeah.
pointed there, right?
No, you have to
You have
point
to point
here.
here. Ah.
This is just for you to know if you
Because
don't
I
want to
thought
look
that
back.
there
I
were
always-
some of them where you point there and it projects there. I don't know. Yeah, that's what. No, these I did. Try.
Okay.
You want to say hello?
We can start, right?
Yeah. We
And then... Okay, so good morning, everybody. It's my pleasure to see so many of you here, and it's my pleasure to open this mini-symposium that I have co-organized with my very dear colleagues, Ute Krengel here and Andreas Breck. And so in today's mini-symposium, it's going to be divided in two parts. In the first part, we will have a presentations by Marta and Gunnar from SciLife Lab about technique itself, its possibilities, and how they set it up. They are a great facility that we are using heavily. And we will also hear from Thermo Fisher and Geol their presentations on the instruments and what is the current state of art in the field. And we will also hear about mass photometry, which is also a very useful technique. In the afternoon, we will have a scientific talks, and we will hear a number of very high-profile international speakers that will talk about their use of CryoEM and CryoET in answering important biological questions. And so without further ado, I will start. And I will just say that you are very welcome to, at least I've heard that from Marta and Gunnar, that you are very welcome to raise your hand and ask questions during their talks. And for those of you that are not here, that are following us on live stream, you can send an email to my address, and you will find it on pages where Minisymposium is announced. And I will try to read your questions at the end of this talk and every other talk. So just send a question, and I will do my best to bring it up. But without further ado, Marta and Gunnar from SciLife Lab are going to introduce themselves and technique to us. So, go ahead.
Thanks, Nicolia. Do you
We
hear?
need to turn it on.
Do you hear? Yes. Okay. Thanks very much for the invitation, Nicolina, Ute, Andrea. And so I'm Marta, and I work at the CryoEM facility in Stockholm. I know I recognize few faces. So now I try to kind of like gather some example we got from our last years of operation. Really, like these are the most recent, and to hopefully give you kind of like an anthology of some a little anthology of CryoEM usage and at least that we had in Sweden recently. So this is what I mean with my title. And basically here we have like a slice through a tomogram, and this is a structure I determined, not from actually from the tomogram, but just like to say that cryo-EM, we often, as structural biologists, we think just of a technique for structure determination, but in reality it goes also beyond that, I would say. And we can think of it as a whole and as a way of really investigate the life from, at least from the cellular level. Actually, some would even say we can do also tissue to the atomic scale. Because basically, if we choose the right cryo-EM technique, we can go through this range of dimension. What techniques can we choose? So the most popular is probably single particle analysis, generally referred as cryo-EM. And, but then we can have electron tomography, so basically where we collect tomograms of, and again here I stick to cryo, but of course one can also do room temperature. And then electron diffraction. And again, here I refer to micro-ED, that is basically diffraction, electron diffraction of 3D crystals. But in reality, we can think also of diffraction of powders or compounds. But let's start with a single particle. And you might have seen this picture. It was actually, I think, made by the head of our department, Martin, and it represented what the evolution of single particle analysis from before 2013 to nowadays. So basically going from what was called blobology to atomic scale, and where we can actually see side chains and atoms. And this is my personal story with that. So from blobology to side chains analysis on a chaperone protein that I really like. And then, but really we can go atomic, right? So in 2020, there were 3 papers that showed atomic structures of apoferritin. And we're really like, one might even say, it's so hard to build models, right, on these, like when you have really single atoms resolution. And this is because eventually, I mean, you know, with an electron microscope, we can see atoms, right? I mean, nanogold particles. And we can even overcome with it, thanks also to the cryo part of it, we can overcome the electron damage, basically, at least to a certain extent. So how, okay, maybe this is super basic for most of you. I wanted to have like a range of comprehension from, so but what is single particle analysis? Is basically, so we have our protein frozen in a vitreous thin layer of ice buffer, and we shoot an electron beam on it, and we can get a picture of 2D projections of our molecule. And then we go into the analysis of these images. And this arrow here, this green arrow, is thicker because it actually contains a lot of operation, a lot of possibilities also to fish, and then I will talk more about that in my research talk, to fish details about our protein structures. And so now I start with the examples, and so what type of details, for example, can you get? And these are examples that come from users to our facility. And for example, the group of Pontus Gordon in Copenhagen and Lund that work on small membrane protein, and they really have generally very, very nice grids. And they can, I mean, with a single particle, you can basically get information about lipids bound, right, to your membranes. So you can really go and get information about unique event, I would say. because it can happen that you can do a lot of classification and you can end up seeing only certain, you can focus on certain region and get a lot of details that you cannot, I mean, you can get many details with X-ray crystallography, but you average at the end on your crystals and you might not see low occupancy regions, or you cannot do focus analysis like we can in single particle. This is another super recent example from the group of Eric Lindell, and this is a GABA receptor, human, and you can see then at this point you can actually by single particle analysis. I mean, I put here an example of the one of the structures that they got. They got multiple states, and here is a video of the model that they have, where you can see all the it's a morphing in between the different states, but actually they are very strong group in molecular dynamics, so they can combine some of the information that they get from the cryo-EM maps with specific molecular dynamics at the atomic full atom dynamics on given regions. I cannot really talk much about the science of each and every one. I just want to show you the capability of cryo-EM. You can, of course, I mean, this is probably more obvious to everyone that you can, that cryo-EM then it's almost unique in the analysis of really huge complexes. And so this instead comes from the group of Alexei Amunds, and they solved the full respiratory complex, and bound to the membrane, and with basically, I mean, you can see the complexity of such an architecture, and the resolution is so high that you can really, again, go into look at lipids, look at, and you can even look at possible dynamics again. This example comes from me, I mean, from Andy and Rosberg. And it's, I'll talk more about this protein later on. Again, I want to point here to the fact that this is an important protein that was almost, I mean, impossible to guess the folding of. And again, it was only possible with cryo-EM. Then there is, you've seen already in in few of the examples, there is I would say also like cryo-EM starts offering, I mean, it's not the final answer, and some information about the dynamical range of our biological systems. So that, for example, here, this is, you might recognize this, the chaperone that I told you I'm in love with. And maybe I go a little bit ahead. So basically, when you start getting different states by cryo-EM, then you can at least start having hypotheses on how a mechanism, what is the mechanism behind the functioning of a given protein. For example, this is a disaggregase, ClIP-B, that takes heavily aggregated proteins and basically threads them through, right? I want to point, and I will talk more about that later in the research talk, to the fact that so we can get a lot of ideas with cryo-EM, basically then still needs to be tested also with other biophysical methods, I would say, or with computational methods for dynamics. But definitely, this is again a unique tool. So think basically when you get a cryo-EM experiment, you get a lot of information out of it. You get the structure, you get the details, you get the overall movements. It's quite fun as well. I mean, it's not a boring task. And then I want to show you also the fact that another example that comes again from the facility and from the group of Luca Jovina at KI. So he actually had a few publications on this subject, and I was involved in is, so this is a protein, this is uromodulin. It's a protein, it's the most abundant protein in our urine. And actually, this is a picture of urine, that's straight. And then you can purify this protein and really get enough to have a high resolution structure. And this is something that you cannot produce it on the lab. You cannot make mutation. You cannot control the glycosylation. I mean, pretty much you can only look at it by EM, right? But then from an EM point, from a cryo-EM point of view, this is also quite challenging because it's a very, very thin polymer, you see? It's unclear, like in the beginning we thought like, oh, this is an helix, but it's an helix with 180 degrees rotation of each monomer. So it's basically a polymer, like a biological polymer. And so it's still, it's also have this very, very specific arrangement. So each one of the different colors is a different monomer, and you see how each monomer has a tail that embraces the two adjacent one. And then clearly they organize in something we can make an estimation of how they would organize in the bigger picture, like in the full length polymer, and then possibly they seem to come together in big bundles that work as Velcro to basically trap the bacteria in our urinary tract. Again, I picked this example once because of the in one case because of the specificity of this special, like the way you produce it, how thin it is, and all this, but also because then it offered a nice opportunity for combination of single particle analysis, AlphaFold 2, and X-ray crystallography. So actually, Luca is a crystallographer, and he had crystals for many, many years that he couldn't phase. Of a bit actually that is not visible in our, basically the arms here that are not visible in our cryo-EM structure. So what, and these are the bits that bind to the bits that binds to the receptor in the bacteria. And so he basically had the crystals, and then eventually he could phase them with the first prediction of AlphaFold when they came out. Actually, I think it was done by the people and at DeepMind before even they published the paper and the results. And then what was then nice is that what we could do is that, so we then took some images of uromodulin polymers in complex with FIM-HL from the bacteria, so the domain of the bacteria that is supposed to bind to uromodulin. And then basically we could, even though it was a very low resolution structure, we could then combine all the data and fit the new crystal structure and put the FIM-HL. And then what is nice is that there was clearly the positioning of given glycan that sugars that are bound to the bacteria. So all this was to give you a hint, I mean, an idea of what we can do with cryo-EM. These are very few examples from our facility. I just picked them because of some logic, maybe not much of a logic, but yeah. So I don't know if you have any curiosity question. If not, I pass. If you remember, there is, so we talked a little bit about single particle, but then there is tomography, right? Tomography, for example, can be very useful. I mean, this is just a test I had done of growing cells into directly on top of cryo-EM grids. And then even though they are probably just plunged, for instance, in the methane, probably they are not crystalline in the body of the cell, but the periphery was good enough so that I could go to the, this case is a fibroblast, and so I could go just to the periphery here and collect tomograms, and actually the tomograms don't look bad at all. I mean, you have all the cytoskeleton with the different, possibly actin and others. Clearly, microtubules are very easily recognizable, ribosomes as well, and so on. So, but what is tomography actually? I mean, I forgot to say, I mean, probably most of you know, so the difference, I mean, single particle, we go and we shoot each time on a new spot, and we take, and then we average all our particles. And here instead, we are fixed on a region of interest, and then we tilt this region of interest, and so this way we can reconstruct a volume. And this volume is often very, very informative. Of course, you cannot do, in order to be able to do that, your area has to be thin enough that is penetrated by the electron beam. So if instead it's too thick, because say like if you're interested on the center of this cell, then instead you need to fib. Fib means basically what we call fib milling. It actually stands for focus ion beam milling. So I'm sure you'll hear more from Rebecca. And it's basically when we remove the excess of material on top or on the bottom of our region of interest. And we are left out with a thin region that we call a lamella, where we can collect images. Now, we don't have this instrumentation in Stockholm, but our colleagues in Umeå, with whom we work basically hand in hand, I mean, we are the same facility, they do have like an old fib milling instrument that they do research with. And so this is an example from the group of Lars Anders Carlson in Umeå. And you see this is infection of viral infection inside a cell. This should play. So here on the left, you see the tomogram. And here on the right, you see the rendering that they have done, like called the segmentation, where each one of these is a virus. You can go farther ahead with this, and you can actually do a similar thing to what is single particle for 2D projection. You can instead do it in volumes. So you pick out your region of interest, your volumes, sub-volumes of interest from your tomogram, and then you average them, and then you get a structure. So it's something that we like to call as like the future of structural biology, where you do structures from the cell, basically, so in situ structures. But just to get started, so this, in order to do that, you need to have sub-tomogram averaging processes and procedures and way of analyzing your data. The sub-tomogram average pipeline, I mean, you can see here an example actually from Dustin in my team. It's like it can be very, very complicated, but it's really a big area for development. And you can actually get quite a lot out of it. Because, for example, in that study by Lars and Selma, they had, for example, they found that they had certain region, certain kind of like membrane enclosed region in the cell with some fibrils inside that they still don't really know what they are. But then Dustin could perform a subtomogram averaging on them, and then they try to fit all possible like known structures or models into this to figure out what it is. It's still unclear. Of course, like ideally, we like to push this resolution, right? Because it is possible to push this resolution. So this is an example from Giulia Zanetti's group in London, and she really showed together, I mean, the group of John Briggs is one very big example of Peiju Zhang of pushing the resolution with subtomogram averaging. This was one of the first examples of the COP2 coating, and you see it's quite nice. I mean, you can get really at least 4 angstrom resolution information that then you can put back into like these big vesicles that she got structure from. So this was basically my examples for the cryo-ET, and I think it's a very fascinating thing. And again, it's all a realm opened with, so once you have an instrument to do cryo single particle analysis, you can do cryo-ET as well, provided you have thin enough sample. And then you can do something else that is electron diffraction. And this is something that, thanks to the presence of a group at Stockholm University, that Jadong Zhu group that is it's been always very strong in electron diffraction of materials. We are lucky basically because they work a lot at our machines and they teach us a lot. And the micro-ED is a technique where basically you have small crystals of protein in this case and 3D dimensional crystal, and you basically collect a diffraction pattern and then you solve a structure like you do in X-ray crystallography. The type of crystals you use are these, is the one that generally you throw away from your crystal tray in X-ray. So again, basically it opens a possibility, CryoEM opens a possibility to use what you cannot use for the focus electron beam like microfocus beam at the synchrotron in X-ray, and still at least try to make some usage of it, right? So, here you see, I mean, these are the crystals. When you look at them at the electron microscope, they are thin enough that the beam can go through. So typically needles and plates work very well, actually. And this is an example from Tim Schulte. And you can see basically, so this is the electron diffraction pattern, and you see the correspondent map. There is another application with electron diffraction that is really cool. This comes from the group of Onyi Shu, also at Stockholm University. Data collected in our cryos. And it's basically, this is a zeolite. So it's an inorganic cage, but with an organic compound that they were interested in inside. And so the fact is that without cryo condition, they couldn't see the organic compound inside. So they needed cryo condition to be able to visualize it. And then, I mean, you see how amazing the resolution is, right? It's 0.7 angstrom, actually, the diffraction spots. And with this, I think I finished with a show of it was a little bit of a gallery of stuff that came out in the last couple of years from our facility. It's really just a tiny portion, but just to show you all the different type of things that are actually that can be looked at on the electron microscope. And if you have any question. No. Yeah.
I have a question, which is kind of more about the organization actually, because you showed, I mean, really fantastic examples of different technologies, and so you have different scientific groups kind of bringing in their biological and technical knowledge, but then I guess there must be some kind of a backline there from the facility actually that kind of provides support in terms of what like the suboptimal all these things which are really
So actually, we will cover all this on the second part of the talk. Yes. So very good introduction. Because actually, this was to show, this is where we are at in Sweden now. But actually, now Gunnar will show you where it all started. And then I can go through more of the, practicalities, let's put it that way. And we can also chat more.
Okay, thank you, Marta. And I checked, there were no questions online, but I just wanted to say it was a fantastic, show of everything that can be done and how technique actually evolved in the last couple of years. All of this or lots of these things were not possible in this breadth, let's say 10 years ago. So it was fantastic to hear that, and let's continue with Gunnar.
Yeah, I hope this is working. Yeah, it sounds like it's good. Hello, I'm Gunnar von Heine. I'm also involved. I was involved with setting up the CryoEM facility at SciLife Lab, as I will tell you. So I thought what I would do is precisely that. I will tell you a little bit about the history, actually, of how it came to be in Sweden, like it is now. And maybe it can be in some way useful to you as well, I hope. Okay, so speaking of CryoEM in Sweden, you have to talk about Scilab Lab, because that's where it all started. That was the whole basis for CryoEM in Sweden. So Scilab Lab, many of you probably know, have seen it or have been there. It's this now quite big infrastructure initiative that started out as a collaboration between the three Stockholm universities, KTH, Stockholm University, and KI, and soon after also Uppsala University joined. So in Stockholm, Scilab Lab is infrastructure, various types of life science, really expensive and technically advanced life science infrastructures that serve the entire country. It's located in these two buildings, Alpha and Gamma, and there is actually Beta that I think you can see just behind here, and possibly will expand into Beta over the next few years, but it's not quite been decided yet. So in Stockholm, everything is collected in these two buildings. They are on the KI campus, but they run jointly by the three universities. In Uppsala, the Scilab Lab infrastructure facilities are spread out across the BMC, the Biomedical Center, but there is actually a small Scilab Lab building just to give it a focus in Uppsala as well. And nowadays there are small nodes in the other major universities in Sweden as well. So it's really a national organization at this point. So the infrastructure is organized in 10 national technology platforms, and you see them here. CryoEM is under bioimaging and molecular structure. That also includes advanced light microscopy and NMR and a few other things. But you also see the others, of course, genomics. It's one of the major sequencing sites in Europe, for instance, is at Scilab Lab. We have bioinformatics. This is also close to my heart because I was involved in setting that up, and that's basically a bioinformatics consulting service, you could say. So scientists apply for support from the bioinformatics platform, and if their proposal is funded, they get, say, 500 or 700 hours of help from a bioinformatician in their project. So this has become very popular. I think there's more than 50 bioinformaticians employed in this platform right now. But it's not only technology platforms. So in the Scilab Lab buildings in Stockholm, there's also research groups intermingled with the platforms. So there is more than 100 research groups now at Scilab Lab in Stockholm. And they, of course, are from the three different universities, but they've been co-localized within this building. And they would work in areas where the technology platforms are relevant to their research, of course. And in these two buildings, there's more than 1,000 people work now, including the staff in the platforms and the research groups. So it's quite a major constellation now, with a national remit, basically. So Scilab Lab, the first kind of sketchy ideas was in the late 2007 or so, where there was a first vague plan for what they at that time called Stockholm Life Science Laboratory, and it was basically some kind of EMBL-like idea between the three Stockholm universities, but there was no funding around. However, in 2008, the government put out a call for what they call strategic research areas that they opened up for the universities to apply for. And the three Stockholm universities, thanks to the incredible work of Mattias Ullén at KTH, who is this amazing person who just can make things out of nothing, basically, come out of nothing. So if it hadn't been for Mattias, the whole vision of Scilab Lab would not have been around, and certainly nobody else but him could have brought this to fruition in such a short time. So he convinced the three universities to apply together to this strategic research area call with only one project in the life sciences, and that was Scilab Lab, basically the research infrastructure idea. Initially focused on Stockholm, turned out that Uppsala had a similar idea, and they were also funded within the strategic research area. So in 2010, there were essentially two Scilab Lab projects starting up, one in Stockholm, one in Uppsala. And then, thanks to some political help from high levels, it became possible to merge these two projects with extra funding and to actually form a national infrastructure that started in 2012 under the leadership of Matthias, and I was working in this the first 5 years or so to get this going. So that was the organizational background to the CryoEM. But of course, in 2012, nobody knew about CryoEM. Certainly, we didn't. But that was to change. And interestingly, looking back, you could say that the roots for modern CryoEM in Sweden was this workshop in Nagoya, or symposium in Nagoya, in January 2013. And it was actually to celebrate, I think, the 65th birthday of Yoshi Fuyoshi, who was one of the pioneers in cryo-EM. And for some years, he had the best microscope in the world that he built himself together with engineers from Geol. So everybody went there with their samples. But of course, this was still blobology times, as Marta said, right? So basically, it brought together all the leading structural biology cryo-EM people, like Richard Henderson, Nigel Unwin, Wolfgang Baumeister, and also a number of membrane protein crystallographers like Rod McKinnon and Brian Kubilka and a few others, right? To talk about membrane protein structural biology, but within the context of cryo-EM mostly. And then there was one person at this workshop who was not a structural biologist, and that was me. And I was invited only because I worked on membrane protein biogenesis, but I had never done structural biology at all. So this was, for me, a very interesting workshop. The first 2 days everybody was a little bit low because they talked mostly about two-dimensional crystallography, membrane protein crystallography. That was the vogue in these days. Richard Henderson had worked on this, for instance, where basically you crystallize in a plane, you get these very thin crystals, and you put them in the microscope, and you take pictures and diffraction patterns, and from that you can reconstruct the structure. So for a while, that was thought to be the technique that would solve the membrane protein structure determination problem. But it became very clear during this workshop that this was not going to be. So there were a few reports of, decent structures done by this, but there was no real excitement anymore in this. It was pretty obvious that this would be a niche technique. And the rest of it was blobology, until the last day, Yifan Sheng showed the first data obtained with the first direct electron detector on a membrane protein, single particle membrane protein study. And he showed a structure at about 3 angstrom resolution or something like that of an ion channel. And then suddenly the whole atmosphere changed, and everybody was saying, wow, maybe this thing that Richard Henderson talked about 15 years earlier, that you could do high-resolution structure with, potentially do high-resolution structure with cryo-EM, was actually possible. So everybody went away on a very high note, and to me it was great, this sounded interesting, but it wasn't my field, right? But then I spent, as I've done a couple of times before, I spent a month with Rod McKinnon at Rockefeller University in November the same year. And when I was there, I realized that he, who was really a dyed-in-the-wool crystallographer, right, was very busy trying to bring cryo-EM to Rockefeller. So then I started thinking, maybe there is something about this cryo-EM thing anyway. So, and that became even more clear when the Yifan Sheng structure was published in December that same year. So when I came back to Stockholm, I mentioned this at the SciLife Lab context, and Matthias said, that sounds like something we should have at SciLife Lab. Wouldn't that be a good thing? And so we talked to Göran Sandberg, who is head of the Wallenberg Foundation in Stockholm, which is in Sweden, which is this big private foundation. And he said, yeah, let me talk to my scientific advisory board. So he spoke to these 5 or 10 Nobel laureates that is on his advisory board, and they said, yeah, that sounds like a good thing. So he came back and said, write an application. We'll try to fund this. So I was asked to write an application. I don't think I've ever even seen an electron microscope in real life at the time, but so I had to learn quickly. But I got good help. And in the summer of 2014, we together with Umeå, we put together this application to the Wallenberg Foundation. And they liked it, so they very quickly awarded the grant. so we could start procurement of the first microscopes in Stockholm and Umeå. Even in late 2014, we had to recruit all the personnel. There was nobody in Sweden that could do this. So Marta was one of the first recruits, and a couple of other people. We had to prepare rooms. We had to learn everything about, what it's like to have, what the requirements are, etc., etc. The microscopes were actually installed in March 2016. So it was pretty quick, actually, from basically from early 2014 until we had a working microscope set up in Stockholm, and then a little bit later in Umeå. So from the summer of 2016, we could actually start taking users in the facility in Stockholm. And from there, that's what it's where it started. What we have now is like Marta told you, we do single particle, we do cryo tomography, we do micro-ED. This is the equipment we have right now. We have 3 cryosys, 2 in Stockholm, 1 in Umeå. We have 2 200K microscopes, 1 in Stockholm, 1 in Umeå for screening mostly. They all have modern detectors, so they all produce at very high rates. What is it, 20 tera per day or something?
Yeah.
It's a lot of data. They run 24/7, year round. In Umeå, there is a now aging FIBSEM, but this needs to be updated. I think it's really getting old now. We don't have one in Stockholm yet, but we hope to get one. And we have, of course, CryoClem equipment in Umeå, and Marta is just procuring one set up in Stockholm. So if we can also get FIBSEM, we will have this side covered as well. So that's what the equipment looks like right now. And of course, personnel is the key thing here. So this is the people in Solna at Scilaf Lab Stockholm. Half of those people were basically the first set of recruits, and then there has been other people joining. And here is the crew in Umeå. So it's about 6-7 people on each site. We just had, actually 2 weeks ago, these 2 people join. They're image processing experts, and they will be consultants to users who need help with the processing, downstream processing, essentially. And then we have set up something called the National Screening Network, and this is to get the other universities to raise their level of expertise. So we have people in Uppsala, in Gothenburg and Lund that are experts and that run the local cryo-EMs. So there is one 200K new one in Uppsala. There is one going to be one in Lund, and there is just a small microscope in Gothenburg, but they have the expertise at least. So this is through these people we kind of organize the network, and the idea is that most of the screening should be done locally, and then the grids should be sent to the main sites for high volume data collection, basically, and processing. And this seems to be working. So we have a very nice working relationship between the different universities now and this community. And finally, there is also, as some of you know, there is a kind of a Nordic network called CryoNet. This is initially a network for facility personnel in the Nordics, or actually Sweden and Denmark initially, because it's funded by the Wallenberg and the Novo Nordisk foundations. But we tried to extend it to include facility personnel in Norway and Finland as well. So this is to basically to exchange expertise and advice between the different sites. CryoNet also organizes an annual symposium. We just had one in Stockholm. Maybe some of you were there. And this has been growing. We were 200 people in Stockholm now last week. Next one will be in Aarhus next fall, in October probably. And so this is trying to organize the cryo-EM community on the Nordic scale. And I think that's where I should hand over to Marta to tell you more about the details of how we actually run the facility itself.
May I ask?
Yes.
So a very interesting beautiful life science building
Mhm.
built, and it's a cryo-EM facility is planned in there. So I would like to know how. Yeah, maybe I should take the microphone. So I would be interested in knowing how it started. I mean, you had probably only few users in the beginning, but now I see you have all these microscopes there. Can you tell a little bit more about that?
So the way we tried to start it was because, again, we didn't have anybody who really knew cryo-EM in Sweden, meaning we didn't have any users, right? And so we actually recruited also a scientific group leader as part of the package, who was not involved with the facility as such, but who would come in and to do the science. And it didn't take very long for the X-ray people in Sweden, especially in Stockholm, to convert to cryo-EM. So basically, I think you can say over the past 5 years, essentially every crystallography group in Sweden has converted to cryo-EM as their first choice of technique. And they do crystallography only when they have to, so to speak. So that worked fairly well. And
can
of course,
I add? I think actually, I don't know what you did before, but there was a lot of expectation already from other groups. So for example, the whole of the Uppsala crystallographers were kind of like in the queue waiting to enter. So they were among the first users like in March 2016. And they had also like very suited specimen for it.
They worked on ribosomes, so that was perfect.
Or they had like these big viruses, or they had crystal clean proteins. So I would say that, I don't know, there was already quite a lot of expectation in the country.
And then I have another question to both of you regarding micro-ED, because I remember when first reading about there was this beautiful papers about how to use it for pharmaceutical drugs. I mean, they just put a powder essentially on the grids, and then they could find out what was in there, what the impurities was. So what kind of equipment do you need for micro-ED compared to others?
Actually, you need less, I would say. So that's a good aspect of it, because in principle you could even do it on a lab 6 source microscope, even though, I mean, it does help if you have a FEG. Like if you have a good microscope, it will help because you can, because you can control more the beam, it's more stable. And actually, so in Stockholm, Jadong's group, they are developing and they run all the experiment in our machine, this serial ED, so that you can basically move the beam on top of your powder or your protein, whatever diffracts. But on a way, it's kind of like, you need the same equipment that you need for single particle. It is important, I think it does help a lot to have an autoloader, because, for example, the people at Stockholm University that do mainly material, they do have like a very good microscope at Themis and they have like a very good gel instrumentation as well to do their studies. But eventually they do all the compound and anything that is organic, they prefer to do it in our cryos, because because the fact of having the autoloader makes them possible to analyze more
So you say we
sample.
wouldn't need any extra equipment
No,
if we
absolutely not. No.
Yes.
Only you need probably a camera. It's better to have a camera you need a camera with a good, basically dynamical range. And now we have a Zeta D, it's okay. Ideally, you'd like to also try other cameras, I would say.
Thank you.
Yes, but.
So, can I have a question? I'm
Absolutely.
sure I can.
And then Marta should have 5 minutes to tell you about how we actually organize the daily work. But
So my question is more of a practical character, actually. So you obviously have been in actually organizing this from the start. I would really like to know actually how much money went into building this infrastructure?
Ah.
And to kind of an idea, actually, it's important in a way because we...
The whole
we,
SciLife lab, I
yeah.
don't actually know, but that's
No, but
that's
like
big
the
money.
EM
The
infrastructure.
EM, I was just looking, I think from the Wallenberg we have received, and this is for instruments only,
Yeah.
maybe 150 million,
Yes.
something like that.
Oh,
For the equipment, yeah, for both
the
sides.
Yeah.
But that's from the
maybe-
Wallenberg. But then
yeah.
there's been a couple of other
Foundations.
foundations as well. So say 200 million,
Yeah,
maybe.
okay, but that kind of covers obviously quite a
That,
range
that,
of...
that basically covered the equipment. Then, you know, the room cost 5, 6, 7, 8 million
Yeah.
to fix up with all the ventilation and temperature control and
Yes.
stuff. But you're building it anyway, right?
Yeah,
So,
Yes, we do.
yeah.
and then of course it's staff, right? The staff costs, and
And
not to
service.
forget service costs.
Yes.
Thermo Fisher, yes. Service costs are really expensive.
Yeah.
So that's something you have to factor in when you buy
And
machines,
especially
right?
to to factor in as much as possible in the beginning.
And you
I would
need
say.
it because you know you need engineers to come, essentially to be on site.
You made the budget.
Yeah.
Yeah, I would actually just also like to follow up. I think, thank you so much for this historic perspective, because this is something for us to learn from, because we also want to, catch up with all the developments. And a couple of things that were fascinating for me is that you have started, and basically the facility itself has then attracted and developed research around it, but you
Yeah.
started with bare bones, you know.
Yeah.
And another thing that I think it's worthwhile noticing, that you were brave enough, and government and funding organizations, to start in 2 sites at the same time, almost, right?
Yeah.
So I think that this is something, and it's still going on strong in both sides, and actually you are picking up more scientists from around the country that were not in this site. So I think those are all very
So I can just
useful.
make one comment, and I think it's actually because we started this whole thing from scratch, right? And we started it as a national facility from day one. That's how we built it. And I think that has actually made it easier than if there had been, 3 different groups in 3 different universities trying to come together and make some sort of, let's see if we can do something national, but we're really more interested in our own local stuff, right? So, so I think it really helped us to have this SciLife Lab organization behind us, so that it was from day one, it was a national facility, right? It wasn't local facilities trying to get some extra money by joining forces, right? And I think that has helped us.
Yeah, great story. And then, we will have to move forward, but actually, so questions are a little bit lagging behind because people are sending emails. But the good news is that we have been, we are watched like outside of this auditorium, which is a good sign always, right? But so I'll read some, we had...
I mean, maybe we can use
It
the
was,
slide to answer the question. I mean, we have some slide about the actual operation, but like really practicalities, but we can use them to answer the question. Is probably more interesting.
Right, like let's just make sure that we answer all the questions, because that's why we are here. We are here to, to serve the community, right? And so the first question was actually for you, Marta, that came, oops, I know I needed something else here. That came from Ayla, and she's asking, what are the size limitation of the electron microscopes you use? Theoretically, you can see individual atoms, but I've heard that this is very difficult for protein molecules with the instruments available. Why is this?
Yeah, I mean, she's... can you
sorry.
hear me? Yeah, I mean, she's right in a way, because that example from 2020, that's kind of like the lysozyme of cryo-EM. So it's apoferritin, it's pushed in all possible angles, like the FAG is a cold FAG, the detector is is the best one, and use like trying to fish out all possible details. The sample is the most ordered and with high symmetry ever. So she's actually right, you are limited in that. I have to say though, in terms of diffraction, this is quite amazing. Maybe not for proteins, but definitely for zeolite. Like 2 days ago, we saw diffraction at 0.28 angstrom. I was like, this is subatomic. I mean, so basically, you cannot, we don't have a model to build. You need a theoretical model to be able to represent it. So it was like super cool in that sense. But so on a way, I think that the question is actually quite correct. The limitation at a certain point is not the machine, at least with this machine that we have, even though, for example, we don't have the latest PEG. And still, I don't think we are limited by the instrumentation, but we are limited by the sample. At the end, the sample, in order to achieve high resolution, you need to have high symmetry, it has to be rock solid, and you have to dig out all possible details in image analysis. The question now is, are we really so interested in going really atomic or subatomic for protein, or we are more interested in knowing interaction, motion? That's still, I mean, basically you have these two possibilities open with EM. Maybe both have their challenges. Yeah.
Right, and this is actually another very good point that I wanted to build upon. Like when you created your facilities and brought up the instruments, the first one that were interested were people looking at the atomic structures and X-ray crystallographers. But that is slowly changing, I think, because now, as Marta has pointed out, it's not so much anymore about seeing the high resolution. We kind of have the means to do that, and there's only so much we can learn from it. But it's looking at the more complex and more complex processes going within the cell and with so many different partners in the cells that are involved. And I think that's the real, like next revolution that this technology is going to bring up. But there is one more question that I want to read for you, and this is coming from Ryan Osland, and Ryan is saying, hi Gunnar, thanks for describing the powerful and potential national infrastructure for cryo-EM. I suppose this also requires a substantial amount of projects with funding to use the facilities. Could you say something about how the Swedish research funding landscape allows for such projects, and importantly, following the stream of excellent results generated with the facilities, do the funders respond? It asked, are there prospects in Sweden for further growth in this field?
So we have been able to keep user fees very low so far. They're going to increase a bit, but I don't think, we'll obviously we would not be able to charge the full cost, so it has to be covered to individual users. It's growing slowly, but it's still cheap. So it's not a big burden on a project grant at this point to pay for the cryo-EM.
No, actually. I mean, detergents are more expensive. Yes.
But obviously this has been, much... people know about this. I mean, funders know about this as well. And of course, the Wallenberg Foundation has been very, continued to be very helpful in when we wanted to expand and when we wanted to upgrade and so on. And the SciLab Lab organization as well, they pay most of the salaries, etc. And they also see this as one of their more successful platforms. So the Research Council, they fund these smaller research grants, right? Individual project grants. And they're not actually funding the cryo-EM. Yeah. They're not helping to fund cryo-EM at this point. Maybe they can help fund some sort of, we hope, some kind of more community supporting things. But the expensive stuff we cannot get out of the of the public funding in Sweden at this point. This would be quite difficult.
Maybe also another aspect for the question is that I don't think that the projects needs to be there from start. At least in my opinion, at least what I observed is that the project came up. I mean, they just, I don't know, they exponentially grow some way.
Yeah.
It's really strange. But it's really like I was used to the UK where pretty much, I mean, many people were doing EM for a long time. So when I came to Sweden, I was like, okay, I mean, it didn't seem like they were suited projects. But then over the years, already in the first couple of years, they just grow enormously. So I don't think that is good also to just look at how the situation is. It's rather thinking of like for each and every researcher, oh, what could I do with that? Because then it's guaranteed that there's more.
Just a quick follow-up actually on the funding situation. And the so your instrumentation is placed at the universities around. So what's the contribution by the universities actually into this?
Yeah, so the universities in our case basically contribute to some salary costs. They haven't paid for SciLife Lab paid for the labs, you know, the rooms, etc. The Wallenberg Foundation has paid for the instruments, and the university, Stockholm University, has been paying for a couple of salaries, which, you know, is a long-term commitment, and you know, and I think in Umeå it's similar. They're part of a kind of a core facility there, so I think there is some, but they get salary funding from the universities. We're now actually seeing if we can get some money from the universities to help cover service costs as well, to keep you know, to keep user costs down. But the universities are they are putting money in, but they're not the big funder, I would say.
Okay, thank you so much.
In Uppsala, the university bought the 200K microscope, and I think they will in Lund as well. So that's their contribution.
So for the smaller microscopes, not for the very expensive ones, for the smaller university and national funding. Yes.
Could you say something about the user cost?
Yeah.
It's now, you can say, you know it.
It's been 5,000 crown per 24 hours until this October, where we had to raise it to 8,000. But it's still quite cheap compared. So what we did was like to estimate the real cost, which actually lies around 50,000. So basically the user were paying a tenth of it.
Per day.
Per day, yeah. And then see how much we could cover with other fundings, basically, and then the difference, that's what we were charging the users. And now we need to raise it a bit, also because our service contracts are run out, and we have to, to cover.
But it's not prohibitive for users, I think that's the point.
No.
And the initial investments, after having initial investments and then university paying for the staff, it's manageable. We are kind of running out of time, but I think this is so important that if you have a couple of more slides to show, there's something that we didn't cover, maybe you can do it quickly.
I don't know if you wish. I mean, I just thought like I mean, many of you probably know how we operate because you've been. Actually, we realized that you used 15%. We said, or you will be allocated 15% of the time.
Yeah, we used. So we are. As in Norway, we have a proposal for using time at SciLife Lab, and we use yearly between 15 and 30%. I think, like lately, we've been ramping up our usage.
Up, exactly. So clearly, also here, there's more and more
there is more
projects,
and more
right?
interest. Yes, yes.
So it's so.
But I can just add to that. So we have opened up for Nordic users, but we have to be a little bit careful because the Wallenberg Foundation, in their statutes, say that their money should go to Swedish research. So, you know, we kind of take.
We take that decision.
But so we cannot expand too much on the non-Swedish usage.
Yeah, and thank you for that point. So it means that you're really not an international facility, you're really a national facility mainly. And then you were kind enough to us, because we don't have our own instruments and our community is still small enough that you can afford us under your roof. But I hope that our community is also going to grow, and that's why I hope we will be able to establish something in Norway.
It's growing, right?
Yes, it's definitely growing.
So, so with the way we operate, basically we have an entry point for, so this is our web page. So basically we have a portal where people can register and apply, and there are two types of application. One is a bug, so block allocation group application, similar to the synchrotron ones. And these we evaluate them once a year. When I say we, actually it's not we, because we only, so we take the application and we read and check for the feasibility in among facility personnel. But then actually there is a group of faculty, a group of 10 people from that were chosen by the decans, right? So they were chosen. Yeah, so each individual university had chosen representatives that they would sit in this panel that analyzes the scientific and prioritize scientifically these applications, basically. And so we do it on this bug system because it's a bit easier, so that basically, say, like Uppsala University has a bug, Lund another one, Gothenburg another one, and so on. And then these, at a certain point, we felt like, okay, we can also have minor bugs for Norway and Finland, but yes, we had to keep it smaller than the Swedish one, especially now that we are running out of time, on the scopes. But so there is, so basically, generally, the user either, if they don't if users are not part of a bug and they don't know anything about that, then they can, of course, contact us and we will set up a meeting, but they can also go through a rapid access application. Then, basically, if, what we do, rapid access is constantly evaluated. Back, I explained, it's like there is this national prioritization committee. And then basically this way, the users will distribute their time within a bug, which is quite nice because it actually fostered a lot of communication in between the groups at the different university that they have to talk to each other, organize, and divide their time, basically. So it fosters also scientific communication. What we offer is basically we do sample preparation for people, for those that don't have sample preparation capabilities at their sites. But as you saw, we are trying to pass this part more to the local nodes because it's very time consuming, and also it's not worth using high-end microscopes to screen. Especially initially, we helped a lot with that as well, and we are still happy to do it if there is no other choice, right, for people. But hopefully there will be. Data, and then in data analysis, unfortunately, this is a little bit our bottleneck in the sense that we don't have, we have the knowledge, but we don't have the time to help in processing, and also we don't have the computer capabilities. So we are trying to make in such a way that we'll make more usage of the national computational infrastructure. And again, there is a big investment in that sense also from the Development Foundation in what is called data-driven life science, where there's some space also for us, for example, in the form of these two new people that Gunnar showed in the chart of the personnel that are going to help users with image processing using national available computational resources. But then we try to help people from the beginning with what we call a drop-in. So basically every 15 days, people can book 1 hour on Zoom with any of us, and we go through their image processing, we discuss really their case. and then we give suggestion and we try again. Or we also, we do automated data processing. This is when data are coming out of the microscope, but this is never really enough. And then we also have every 15 days, we also have a Zoom group meeting with all the users. I mean, it's not all, there's generally 20 to 30 people that participate, where each one can show an example of something that is working or not working. It's basically a discussion platform for image processing, mainly. And then we organize courses, roughly 4 or 5 a year, we've calculated, going from basic EM, so for which there are 2 courses per year in Umeå, to advanced data analysis that we used to run in Stockholm every year, but now we are actually join forces with the Danish people within CryoET, so that we can run really an advanced EMBO-style course for image processing. So we had the first course last year in Aarhus, and next year will be in Stockholm, 2024. And then we developed, it was mainly Jose Miguel's in our team, now unfortunately left, but he brought this project with him to new places. And it's basically, this EM Hub is a platform some of you might know, where you can enter and book time on the machine directly as a user. You can log your information about your project so that you have kind of like a lab book of a given data analysis, or not data analysis, rather data collection, and where you store your sample and so on. And this is opened to download on GitHub, and you can adapt to your own needs as well. And that's all.
Okay, thank you so much. This was really, really useful in so many ways, and there are many things we can discuss about. And we went a little bit over time, but I think it was all for a good reason. Now we are going to, now we are going to hear a little bit about the instruments directly from Thermo Fisher. So Rebecca, the floor is yours.
Wonderful, thank you very much. Is it? Nobody can hear me.
How about now?
No, how about now? Yep, yay, okay, excellent. Well, that was really a fantastic and very inspiring journey through the history of cryo-electron microscopy in Sweden. And I think it's a real pleasure to be here at this moment in Norway. This is my first time in Norway, and I think you as well as a community are at a very exciting stage, thinking about the next steps for you in terms of electron microscopy infrastructure. So thank you very much for the opportunity to come and talk to you today. So my name's Rebecca, and essentially, so I moved to Thermo Fisher 5 months ago now. Prior to that, I was at the University of Leeds working with René, so I'm looking forward to his talk later, where I managed a cryo-electron microscopy facility with 2 Titan Cryos instruments and 3 other transmission electron microscopes for 8 years. And I really fell in love with electron microscopy during my PhD as a life scientist, as a biological researcher. And the reason is there is no other technique that allows you to get such a range of information on such a range of different biological specimens from one instrument. And I was absolutely fascinated and captivated by that. We've already heard from Marta some beautiful examples from of some of the structural and single particle work that can be done, where you can get these amazing high resolution structures. And I'll talk more about that in my talk later today. But we can also be using the same machine as chemists to be looking at crystals of small molecules, as cell biologists who are interested in studying ultrastructure, and even moving up into tissues and even organisms, C. elegans, for example. You can be using electron microscopy to be getting powerful imaging data from a whole range of different samples. So hopefully I'm going to cover a few of those different ones today. So I thought it would be interesting to start off with a little overview of just where electron microscopy is in the Nordics at the moment. And when we mapped this out, actually there are a very, very large number of systems in the region at the moment, and it's been fantastic to see some of the outputs coming from the SciLife labs. But in terms of the map, where we are at the moment and today, you obviously have the Talos F200C, and I was learning yesterday about some of the fantastic research projects you have coming through this. But in the Nordics, Norway is the only country today which doesn't have a dedicated cryo-electron microscopy system. And given the depth and breadth of research that you have within the country, and obviously the demands for cryo-EM in the region, I think it's obvious that, that there are a large number of research projects that could benefit from this technology. And actually, I did a little bit digging into the data, and I thought you might like to see these graphs. So this was me in my hotel room last night make it on some spreadsheets. So we can have a look in these different countries. Different countries have different number of instruments. And we can look at how many structures, so how many publicly available structures are deposited into the protein data bank. And you can essentially draw a ratio to say, okay, well, based on each, in this case I did it on Cryos microscope, how many structures are coming out. And I think these trends, these graphs show are really, I think that supports what Martha was saying about when instrumentation goes in, research projects evolve and come online, and then you start generating a lot of structures. So last year Denmark had 40 structures per Cryos installation, and in Sweden this was 47. Some papers produce very large numbers of structures, so I was going to ask Marta later what we think was causing the spikes, but we can go and have a look. If we have a look at these Nordic instruments and then essentially the output from these are more efficient in terms of the installations themselves than some of the countries in Europe, which you think of as being real cryo-EM and are real cryo-EM powerhouses. So if we look at the ratios for Germany and for the UK, then we can see that these Nordic installations are incredibly efficient in terms of their output. So massive kudos to you as a community for making running instrumentation in such an efficient way and getting such outputs. So in my talk today, what I'd like to do is delve a little bit more into the ecosystem of hardware that's available, and I guess show you some what I think of as really cool stories and applications for these. So at the moment in Norway, you have a Talos F200C, and as I say, this is a very versatile machine, and but it is ultimately a side entry machine. So it's not dedicated for cryo-electron microscopy. It's used for a whole range of different purposes. And today I'm really going to focus in on the Glacios 2 and some capabilities there. So Marta has really set me up beautifully for this, so thank you very much. So first of all, I'd like to talk to you more about the high-resolution structural biology workflows. So can I just do a quick hands up? Can you put your hands up if you consider yourself a proteins person? So if you work predominantly on proteins, excellent. And can you pop your hands down and pop your hands up if you consider yourself to be more of a cell tissue person? Okay, that was the worst half hand I've ever seen in my life. Okay, so that's a really good mix in the room. So the first half is more of the proteins, and the second half will be more for the cell people, I promise. So a little bit for everybody. Single particle cryo-electron microscopy is a growing technique. Obviously, we know there are a range of different techniques available to structural biologists if you want to understand the structure of a protein or a macromolecule. And we're expecting this year that cryo-electron microscopy will overtake every other technique, including X-ray crystallography, as the predominant technique for determining structures of proteins, which I think is a really incredible milestone for the community. And I think it's really interesting to reflect back on the history of the hardware and the hardware development. So I focused in on the Glacios 2 here, and if we go all the way back to 2017 now, for apoferritin, which is the world's most boring test specimen, but for apoferritin, it's a good test specimen for us to test the microscopes, so rock solid protein. It was taking us 30 hours to get to about 3.3 angstroms resolution. And back in 2017, that was pretty cool because this is a 200 kV instrument, and we were going, wow, this is amazing. If we fast forward today with hardware and software developments that I'm going to talk to, on this particular instrument, we're getting structures on this Glacios 2 of 1.6 angstroms in 7 hours of data. So we've seen both an improvement in the data quality and also the quantity of data that we can collect, for example, in an hour on our instruments. So in terms of the system itself, the Glacios 2 is an incredibly flexible machine, and I'm going to delve into some of these details. But as a transmission electron microscope, you can, in terms of a range of research applications, we can work on everything from cells. This is a bacterial cell example. Proteins, macromolecules, crystals. So it's an incredibly flexible machine. And some of the latest hardware and... I'm wandering too far away. Some of the latest hardware and software... No, it's not working, is it? Let's go with that. No. Did I switch it off? Did it die? No, it is on. The laser pointer still works.
Is it going to work?
It will work with that. It'll be... I will switch to there. Ooh.
But it's not.
Oh, is the laptop
It's your
frozen?
computer.
Oh no. Well, that's never happened before. Okay, sorry, bear with me a second and we will just... and we will just... Yeah, exactly. Yeah, that's just completely frozen. Let's try closing it down and opening it up again. Nope. I'm gonna smother it. I'm just gonna kill the laptop. No, even that is even smothering it isn't working. It's like it just won't die. Sorry, this isn't gonna help the timings either, is it? Okay. Right. While that one is coming up. Yeah, it's just while that one exactly. It's never died. It's never died before. It's interesting. No, it should be okay. It's just restarting again. So while that's starting, I'm going to talk to you about what my slides were going to say. So the Glacios 2 has a whole series of different things on it which really make data acquisition efficient and allow you to collect the best quality data possible. And for me, we've seen lots of hardware developments over the last few years, and these include spatial recognition. So I'm just... Oh no, I'm still thinking about it. So for me, some of the hardware developments have been really exciting. So these include things like, in the last couple of years, we've had a new, essentially optical modifications to our column that allow us to collect more data for every area of the grid that we're interested in. And this is something called fringe-free imaging. And we've also had a new series of direct electron detectors and energy filters come online. And these allow us to collect incredibly high quality data. There we go. And these allow us to collect incredibly high quality data. But I think for me, if we look at the hardware and the software available, some of the biggest advances have really been made around software and automation. So we're now at a point where so one of the big step changes between the Talos instrument that you have on site here today and the Glacios instrument, which is there we go. Sorry, apologies. Okay. So. Okay, looks like we're back in business. Okay, here we go. Wonderful. Okay, and we're working. Okay, excellent. Sorry, apologies for that intermission. As I was saying, in terms of the Glacios, we've got hardware and software modifications on the instrument or features on the instrumentation that allow us to get more out of every unit of access. So the first one that I wanted to highlight was really this fringe-free imaging. So this came online really quite a few years ago now and is routine on all of our cryo-electron microscopes from Thermo Fisher and has been for some years. So we can now modify the optics to allow you to achieve this what we call fringe-free illumination. And the take-home is that it basically means that you can squeeze more images into any given area of interest and get a higher throughput in terms of your data acquisition. The second hardware modification that I wanted to highlight was around the energy filters. And I think these are really, really powerful tools for us to be getting a better signal-to-noise ratio in our image. And the new Selectris direct electron detector from Thermo Fisher, so previously most facilities had a third-party energy filter on their system, and that worked quite well, but there were maybe some stability issues and also some software integration issues. And the Selectris really comes in and allows you to collect incredibly beautiful data routinely with small slit widths, which basically allows you to collect data with better signal to noise, really routinely. So these are just some small hardware features, but as I said, software is really one of the areas where we've seen huge increases. So we now have this smart EPU package, which contains a number of features, which basically allow you to get more out of any given hour on the instrument. So when I first started managing the facility back in Leeds, we were running sessions routinely that were 24, 48, or 72 hours long. So we would load a grid, we would collect the data for that duration, and then the user would then go away and process it. And what we're finding now is that the direct electron detectors and the software are so fast and so good, you might be able to collect enough data on your specimen in just 15 minutes, maybe an hour of data acquisition. And this presents a real challenge for facilities in terms of how you get the most out of your instrument, because obviously facility staff need to sleep, for example. And this is generally recommended. So this is where I get really excited about some of these new software tools, such as EPU Multigrid. So this is a piece of software that allows you, in just a few clicks, to take 12 samples, which are loaded into your microscope. You can set up an experiment that says, I want you to collect this data with these properties on these different grids, and then you can press go and have a lovely dinner, go and sleep and be a human being, come back in the morning to data on a range of different samples. So I think it's great to see the software and the pace of improvement in the software keeping up with the improvements that we've seen in the hardware. So, too much clicking now. So I wanted to tell you a short science story. So this is from Basil Graeber's group. And I really love this as a case study, because Basil was really interested in this complex here, which is CDK7. It's a master regulator of cell growth and division. Obviously, in the title, you can hear this is probably going to be of interest to cancer biologists and people looking at developing therapeutics in cancer settings. The problem is it's an incredibly tricky target. So a large number of clinical trials on inhibitors for this family have been abandoned. And the reason for that is that there are a very large number of structural homologs, so very close structural homologs. And there was just a big lack of structural information available. So in order to enable better drug discovery and inhibitor design for this family of complexes, they wanted to try cryo-electron microscopy. And I think it's worth saying this is an incredibly challenging sample. It's small, it's got no symmetry, and it's really at the small end of what generally you would work with in terms of a protein or macromolecule. I think it's 80 kilodaltons from the top of off the top of my head. So we've got this very small sample. And what Basil was able to do was make grids. They made grids with 14 different inhibitors. They made a duplicate of each grid, and then they collected a small data set on a Glacios instruments for each of these grids. And in their first session, they basically had 24 hours, I think it was, on the instrument, just less than. And they were able to use the combination of tools I've described to solve 24 structures in 24 hours. And these 24 structures of this very challenging biological specimen were being, or what we would have traditionally considered to be incredibly challenging, were down to, were very respectable resolutions, 3.5 to 4.5 global angstroms in global resolution. And this little purple dot here is our inhibitor density for each of these. So immediately they were able to see small molecule binding, for example. And then with this, and I think, one of the other really wonderful things about electron microscopy and cryo-electron microscopy, and one of the reasons that this project progressed so quickly is although there was very little structural work that had been done previously, there was an awful lot of biophysical characterization. And this is where I think electron microscopy being used in conjunction with other techniques, mass spectrometry, we're going to hear a talk later about REFINE, which I'm really excited about, REFINE mass photometry, which I'm really excited about as a biophysical technique that can be used in conjunction with CryoEM. I think having all of that information then allowed them to be incredibly efficient when it came to the structure determination process. So they obviously took these maps, which were at resolution enough to be able to solve a good structure and see a small molecule, but weren't at sufficient resolution for us to be able to determine, for example, the pose of the inhibitor. So they collected slightly larger data sets, and were able to improve the resolution slightly, and at this point they were able to start saying something about the pose of the inhibitor, for example. They then took the best grids and the ones that they were really interested in solving to higher resolution, went and accessed a Titan Cryos instrument, and they were able to really push the resolution on this. So with 10 hours of data collection on a Cryos, they're able to get to 1.9 angstrom resolution. You know, this is, for any structural biologist, really very beautiful density, and we can see ordered waters. So I think this was a lovely result. And I think it's worth noting at this point that actually if they'd have just collected more data on the Glacios 2, and this is an experiment that we did after the fact for intellectual curiosity, you can use the Glacios 2 to get really toward, you know, you can get nearly the same resolution that you can get with the Cryos. Not quite, but you're getting there. It just takes you a longer amount of time. So I think this is an interesting case study for many reasons. And when Basil published this work, we saw lots of people on Twitter saying, "Hey, you know, this is awesome," and this I think really represents the kind of thing that can be done and the kind of throughput that now you can start achieving with the right projects, which is awesome. So hopefully what I've shown you here is that we've taken an 80 hours of 5 kilodaltons out. So it's 85 kilodalton protein solved to high resolutions and a really interesting project. How are we doing for timing? Because I'm conscious I overran.
You're good, but we're late.
Okay, fine. Excellent. So I was a biologist, and one of the things which I think is amazing about the cellular environment is if you take every single cell, so every single cell that you work on, every cubic micron of that cell has between 2 to 4 million proteins in each one of those average cubic microns. The cellular environment is an incredibly crowded and complex place. And you know, I started as somebody who was interested in cells and then kind of became a structural biologist. And I think we're at this really exciting time now where technologies are starting to come online that with electron microscopy that are now truly linking structural and cellular biology. And I think this is just such an exciting field that I had to include some slides on this for you today. So cryo-electron tomography, which Marta introduced, is showing a similar trend in adoption to single particle electron microscopy, but we think this wave is approximately 8 years behind when we look at the EMDB entries. And when we have a look at cryo-electron tomography, we can see even between 2021 and 2022, there were nearly 2 times the number of depositions in this space. So there are more researchers getting to this all of the time. So I'm going to skip forward these next couple of slides because I was just going to talk about software, but in the interests of time, I'm going to get rid of that. Oh, no, there we go. So in the same way that we have software tools available for structural biology and single particle electron microscopy, we have a similar suite now for cryo-electron tomography, which is making it more accessible than ever. But of course, if we want to work with cells and tissues, as Marta described, sometimes we're not able always to work with plunge frozen tissue because of issues with sample preparation and vitrification. But we can move to be working with high pressure frozen tissue, which gives us excellent preservation of the tissue, but we can't just put that into a transmission electron microscope. The samples are just too thick. So I'm going to skip forward a few slides. So in terms of a technique, cryo-electron tomography is now able to achieve incredibly high resolution. So cryo-electron tomography has always been considered to be a low resolution technique, But with this incredible brick of a protein, apoferritin, if we now put this through the latest electron microscopy hardware, we're able now to get 1.5 angstroms resolution reconstruction of this by cryo-electron tomography. And I think what this demonstrates nicely is, as Marta said, that actually most of the time now, the electron microscopy hardware itself is not limiting for us in terms of the resolution that we can achieve. The limitation is the complexity of our biological systems, and of course that's where that's a very exciting place for us to be. So if we want to image these thicker specimens, these tissues, what we need to do is make that sample thinner. And one of the ways that we can do this is by using what's called a dual beam instrument. So these are scanning electron microscopes, which then come in with a second beam that can blast the sample away. So you can use an SEM to image and then be removing some of the sample, either with a gallium beam on the Aquilos or with plasma beams on the Hydra and the Arctis. And I wanted to share with you just a couple of images from the Hydra system. So the sample that I'm about to show you is from a mouse brain, and this was a collaboration with a customer. And what we're doing here is we're essentially zooming up and down through a block of brain tissue which has been preserved by high pressure freezing. So there has been no chemical fixation, there has been no dehydration. This sample has been preserved in such a way that the molecular organization of this sample is completely preserved. And I'm very excited to be hearing René's talk today, later today, which is going to obviously be focusing on brain tissue as well, I think. So, so I think these are the kinds of images which we can get out of this scanning electron microscope, and they give incredible information and context. And what I think is incredibly powerful about this is we can use this SEM imaging, and at some point that we find our feature of interest, and I don't have time today to talk about the integrative fluorescence microscopy, which we can also bring into these workflows. Once we find our feature of interest, either from the SEM or from our fluorescence instrumentation, we're able then to pause, lift out a chunk of that tissue potentially, or lift out a region of interest, mill that down to a thin lamella, and then put that into the transmission electron microscope, and then we can apply the cryo-electron tomography workflows which I spoke around earlier. So I think this is an incredibly powerful technique, and it's very exciting that we're now at a point where these imaging techniques can really marry the more cellular and even tissue level information with the molecular organization. So I'm going to skip through this slide, but the video I wanted to show you was of a single cell. So this is some, actually, I think I will have to show you on this. So this is similar data on a single cell, on a clammy cell. So what we're doing here is we're essentially milling through a cell and collecting an SEM image on each side. And what I wanted to show you was the kind of rendering and richness of biological information that we can get out of this. So we can use a program called Amira, again, a part of Thermo Fisher, to go through and segment this kind of density to be able to reveal the molecular organization, the ultrastructural organization of this cell. And then we can annotate this and really see on a very, very detailed and volumed basis exactly what is going on there, which I think is incredibly exciting. Okay. So for anybody who works on room temperature specimens, if we go from cryo conditions to room temperature, we're able to access even larger samples again. And there's a technique, which I won't talk about in detail, called spin milling, which allows us to access areas even up to 1 millimeter in diameter, and start to and generate images both in 2 dimensions, montages, but also 3-dimensional volumes on a whole range of different tissues. So this is a beautiful image, I think, of a tobacco leaf. And on the next slide, I think I have an example of some virus infiltrations into cells. So we have a really, we can see here the scale bar here is 5 microns, and we can be collecting a significant amount of data in X, Y, and Z. So I'm really excited about dual beam generally, but Hydra in particular is a very exciting instrument because it has so many applications from room temperature and large volumes right the way down to enabling high resolution cryo-electron tomography of tissues. So with that, I'm going to shut up in a minute. But hopefully, what I've given you a super quick snapshot of today is how electron microscopy can be used as a really powerful tool in the life sciences. And we're very, very excited that we're going to be running a live, so jeopardy might happen, like it did on my presentation today, a live demonstration of the Glacios 2 and a bunch of hardware and software features. That's going to be at 10 a.m. tomorrow, and the room is like a really cool theatre format. So
yeah, I
bring
have
your
to
popcorn.
say this is, just for everybody to notice, this is a change from the original plan. So tomorrow's session is going to be in this building, in the room Shino. That is just around the corner, this same floor. It was scheduled to be in Oslo University Hospital, but for security reasons, you know, we had to reschedule it. However, those of you that have registered to visit the facility in Oslo University Hospital will join Andreas after that, and you will be, shown everything. But the demo is going to be here. Yeah.
Wonderful.
Thank you, Rebecca.
Thanks.
So in the interest of time, we're going to limit questions. So if there are any questions, maybe one question. Okay.
Thank you very much for a very exciting thing. But when we are to install these kind of instruments, how vulnerable are they to the external environment? They're put into vibrations, magnetic fields, etc. What do we need to take into account?
Yeah, so every instrument obviously comes with some pre-installation specifications in terms of magnetic fields, vibrations, temperature ranges, for example, in which they're stable. There have been changes to both the Glacios and the Cryos, the latest generations, to reduce those room requirements. So if you go back to when Marta was installed, or when Sweden were installing, the room requirements would have been higher, and today we can be slightly more flexible. We can offer essentially one-to-one support through that as you're looking to work through the building and the building requirements. And I know we've started conversations, but that's something where, as a manufacturer, obviously we're very keen to make sure that the instrument as it gets in is working in the way that you need it to, and environment is a big part of that. So anything we can do in terms of helping you through that process, we'd be very happy to do.
Shall I just
Yeah.
comment on that too, because obviously I haven't installed it. So we have a 200 kV Talos and installed this, so it's on the 4th floor and it's very close to a tram line, and the tram line has a super heavy field actually. So we installed also magnetic shielding there and active kind of shielding. It works perfectly. So this is not really an issue unless you have some whatever.
Yeah.
High power cable through your room.
There are instruments very close to the London Underground. There are instruments up the very, very high in skyscrapers. There are some very, there are instruments, so there are Titan Cryos instruments installed in Delhi, for example, where they have frequent power disruptions. So there is nothing that cannot be overcome with some planning.
Yeah, I'm just going to comment one thing also here that I was very excited to see that 200 kV instruments are actually which are less demanding in terms of space requirements, but also are cheaper and more affordable, that they are actually reaching these very high resolutions and very high capabilities. And if I'm not mistaken, you can use them also for more like tissue, looking for tomography purposes also, right?
Yeah, absolutely. So everything you can do on a Titan Cryos, you're also able
Yeah.
to do on a Glacius. So that's the 300 kV system versus the 200 kV system. You might not get quite as far, you know, really at that cutting edge, and it might take you longer, but they are close.
Yeah,
and
but
generally,
it's a good starting point,
exactly,
right?
and especially where you have a wider ecosystem where you are accessing cryoses as well. I think it can be, as Marta said, the majority of the complexity of hardware is getting so good, in both 200 kV and 300 kV. Often the limiting factor is the complexity of our biological systems. So, yeah.
Okay.
Which is a nice place to be.
Okay, thank you so much. And now we're gonna hear a presentation from Geol, which also has good quality instruments for the same purposes.
I'll hand you that one over. Yep, thank you. I hope your laptop's better quality than mine.
Let's see. Fingers crossed.
It was him.
So, yeah, thank you and hello. My name is Emanuel from Geo. And yeah, okay, running. So, and I'm going to give you some insights about the CryoEM series and the systems we do provide for the cryo applications. So, okay, just a little bit background, just a few slides also locally here to know with whom you are dealing. So Geol is a rather old company in terms of electron microscopy. First TEM released 47, so we have been around for quite a while. That's one of our first TEMs, so if you have ever had the chance to visit our demo center in Japan, it's just in the lobby in the entry. And yeah, we are placed in Japan, the headquarters in Tokyo, but do have offices all over the world. However, manufacturing is taking place over there. Okay, besides TEMs, which I'm going to talk today about, we have different systems, SEM, EPMA, OG. We have preparation systems, dual beam systems. I'm going to talk a bit about them as well. Semiconductor stuff, additive manufacturing like 3D printers and so forth. And of course, analytical systems like NMR mass spec, so which nicely line up with the other systems. Okay, yeah, so like how do the systems rank within GOL? So smaller TEMs, of course, general application, life science and material science. However, then it diverges into multiple purpose systems like 200 kV FACS systems, and then the cryo-TEM series. So cryo-ARM stands for atomic resolution. The ARM series is for the material market, which is well established and known, and the cryo-TEM will derive from there. Okay, for local organization here, for the Nordics and Norway, so we have a demo center in Stockholm and in Germany. So I'm from Germany, as you see it in the back, and there we do have all the demo instruments and customer support, service points, and the support team, as well as we do provide some analytical service and contract work and in-house courses and seminars. Okay, that's our team. So if you are working with a GOL, you might have seen one or the other from the application team here, which are responsible for this area. It's a wide range coming from like NMR people, physicists, material scientists, chemists, and biologists, as I am. All right, and beside my colleagues here, we have, of course, colleagues in Europe scattered and in Japan. Okay, what does it take to make a cryo-TEM? So we used, for our latest series, the established systems in the material science market and adapted or modified them for their needs in the life science segment. So we have, as we do have a wide installation of TEMs in the material field, we took those experiences, especially in the type of emitter sources and so forth, to adapt them and implement them into the cryo-TEMs. Also mentioned in the talk by Gunnar, so we have a long experience in manufacturing cryo-TEMs, be it under liquid nitrogen as well as liquid helium-cooled systems. And in order to make to accomplish the automation needed, we took our semiconductor equipment, which has all this automation, and implemented this into the cryo systems. Okay, here's just a time scale on the cryo systems. Here's Fujiyoshi sitting on the microscope in the 80s, late 80s, and then it went all the way through until the latest generation of CryoEM, which was released 2018. The first type of CryoEM, so fully automated cryo-TEM, was launched. Okay, now we have installation bases worldwide of around 30 installations since 2018, and yeah, more than 50 worldwide. You can look at this nice cryo map, which shows all the cryo labs worldwide. Okay, now a bit more to the CryoEM in detail. So we have two versions, a 200 kV and a 300 kV version, which differ a little bit in height. That's the most obvious thing if you stand in front of them. However, for the 200 kV system, we offer two choices of pole pieces. So a normal wide gap pole piece, cryo pole piece, which allows for tomography. In addition, we do have a HR, high resolution pole piece for cryo work, which is dedicated to single particle work. And there we actually come to this interesting point: is how useful, or do you really need 300 kV? Well, for tomography, it's reasonable. For SPA, it's one can discuss. Okay, we heard already about the emission source. So GOL is since many years relying on, or we think it's a most useful source, using a cold field emission source. You have a very coherent beam, so you don't heat the tip. The tip runs at room temperature, which gives coherence, means the beam does not scatter that much, if you would say so, and allows you to get a smaller point focused. And this at the end improves the resolution in many aspects. So it makes it simply easier for you to achieve a higher resolution. As we have heard, the sample in life science is still the most critical point. So the TEM can go way higher in resolution, so your sample is limiting. But nevertheless, it helps you to achieve your resolution a little bit easier. Okay, here are just some plots about the stability and the energy spread I mentioned. So in former times, like 20, 30 years ago, cold fags were rather unstable as they run at room temperature and accumulate contaminations on the tip, so you had to flash them frequently to get rid of this. However, with an improved vacuum system, you could drastically minimize the interval for flashing, which is now around 8 hours, at least with our machines. Okay, here's just some easy specs what a system can do if you don't have a cryo sample in your cryo microscope. So, and the difference between a cold fag at 200 and 300 kV and simulated resolutions with CTFs for the different emission sources. So TFAG in red, so a Schottky type, and the cold fag 200 and 300 kV in comparison in magenta and blue. Okay, and here again, comparison of the different systems in total, so like available more or less. So you have 300 kV, you have the 200 kV, the high resolution pole piece, or with a wide gap pole piece, and then the two other systems available, and the spatial frequency plotted. So as you can see, actually 200 kV with a HR system is quite close to a 300 kV classical wide gap pole piece. Okay, this has been published recently, or this year. So what can be achieved on a platinum iridium sample? Just put it in to benchmark your system at a 300 kV, so with a wide gap, so a large gap pole piece and a CS of 2.7, and you see you can easily go to 0.6 angstrom without problem. Okay. We heard in a previous talk about how to improve the throughput. So one of the features you can use is to make a fringeless or minimum fringe illumination, the Köhler mode, to minimize or to increase the usable area of your detector. And with this, you increase, of course, the throughput. And there was a nice publication in 22 where they compared at the Osaka laboratory different instrumentations about throughput and about images achieved per day, so 24 hours, and you see it's quite a bit of an increase over the last 10 years. Okay, what else has been done for the CryoEM series? So users might know that if you toggle between different magnification states, lenses get turned on and off and so forth, and this includes some alignment steps. and those alignment steps might need some time. So to minimize this, a new lens design has been released for the CryoEM series, where you do not switch off the lenses anymore. So it's we have a dedicated objective mini lens, and this needs to be turned on and off. In older systems, then this has been gone now, so you can easily toggle between all low and high magnification ranges without aligning the microscope anymore. Low magnification range is important, of course, as you see here, field of view for different detector types, one view, Apollo, or a K3, and the lower you can go, the less images you need, or the less time you need to consider in terms of acquiring an atlas or a map or whatever. Okay. Now the next feature I want to briefly mention is the spec porter, the autoloader of the GOL microscope. So it's a little bit different from the systems on the market so far, as it's flexible. So it's very much appreciated, especially by pharmaceutical industries, as you can run different projects in parallel. Meaning, you can insert a sample, leave it in, retract all the bad ones, and then collect data sets. And if you want to stop, for example, and another group comes, wants to do tomography, they can load their samples, and you can still leave your samples in the autoloader, which opens up quite a lot of options to run projects in parallel. So the autoloader holds 12 grids, and as said, you load subsequently sets of 4 until you have it, or when loading one, you can actually retrieve the bad one, so you're very flexible in loading and unloading your samples. Okay, so how does it look like? That's the GUI. And as mentioned, you have this magazine holding 4 cartridges, and you can freely select which one to swap and leave in every time you load or unload a sample. You can give it tags, used, protect them, names, and so forth. And of course, we have a dedicated loading mechanism, which has airlocks in between. So that is quite nice, actually, for a very low contamination storage of your samples in the spec porter over many weeks. Okay, how do those samples look like, or how do you load your samples? We use cartridges, and we have 4 types of cartridges. So there's a classical one where you take your 3mm grid and clip it with a clip ring, which is not so commonly used anymore. Most of the users use the recoverable types, where you have a sliding mechanism and just slide with a spring your grid, put it in, slide it, fix it, back and forth. We have 2 versions, the one for a normal grid and the one for an auto grid, so a pre-clipped grid. So you can load to existing machines from other manufacturers. And there is a flip-flop version for dual-axis tomography. So, okay, this you do in the cryo workstation, so fix your grid in the cartridge of your choice, then transfer it to your transfer cup and go to the microscope, and the rest is just a button you have to push. Okay, here a little bit schematic drawing of the clip version, the rotation version, and the like sliding version. So here the type with a pre-clipped grid, you just load to the cartridge, close it, done, that's it. Well, of course, you have to imagine doing this under cryo conditions. And there were a publication where they exactly did this. This has been published last year, where they used Talos system to screen, an Arctica to screen the grid, and then transferred to the GOL for final data collection. And they achieved a below 2 angstrom resolution. Okay, this is a working station, so that's where all the clipping happens. So I will, for the sake of time, skip over this, as it's already like 11:30. But we have a working station, that's your cartridge. You put your grids in, then you put it into the magazine, and the magazine gets transferred to your transfer cup, and then you go to the microscope. Okay, GOL has a long experience in manufacturing energy filters. They always come mounted within the column, and the so-called omega energy filter, as they do have the omega shape. The advantage is they're always pre-aligned and set and nicely shielded within the column, so very, very stable. And in addition, you're very flexible in terms of the camera you mount towards your system. So whenever a new camera gets released, you do not rely on any filter whatsoever, as you just plug and play any camera to the system. Okay, so what you do is clear. You increase the signal-to-noise ratio by getting rid of the inelastic scattered electrons, so that's what an energy filter does in life science, at least. Of course, you can do yields and so forth as well. Okay, that's how it looks like without zero loss and zero loss. Okay, as mentioned, so we provide or we are open to all camera manufacturers, so any type of direct detector or pixelated detector is fine with our ARM series. It depends simply on your choice, or if you combine a direct detector with a pixelated detector and so forth, or classical CMOS type fiber optic coupled camera. Okay, software-wise, we have as a basic platform for data collection CryoEM. However, this is like, if you work with CryoEM, it's a very scientific software, it's nice and powerful, but if you do not have much experience, it's not so sexy looking. And therefore, there's a second level, like for users, real users who just come to collect the data, and there we use SmartScope. This is quite nice. SmartScope is a very nice user interface, which is browser-based, so every user can access it from a browser and get started with the data collection. It gets a direct feedback. It got published last year. And we have now 3 installations where we run SmartScope on the systems, which is very, very nice. So you can direct, so it's on top of CryoEM. It talks to CryoEM, but you don't need to do any scripting and so forth anymore. It gives you direct feedback, so you can access your data in real time. Multitasking, you can remote access, of course, and it's nice for multi-user environment. For a facility, you don't get full access to your system hardware-wise anymore. You just deal with the software as a user and let the power users, administrators, take care of your system. So you have more time for other tasks. And as mentioned, it's very easy to install, so you don't need to do calibrations and so forth. As for CryoEM, this is done by us. We do the basic installation and provide you with the scripts for CryoEM. Okay, that's how it looks like. So you acquire your atlas, you can take squares on your atlas, click on a hole, get direct feedback. There's AI built in, so it judges on quality of your holes and gives you a nice prediction on where you should collect or not. It talks to your autoloader. You can define settings on how you acquire your data, defocus, how many images you want to collect per hole and so forth, so as shown here. All this is implemented as well as like focus settings and so forth. Yeah, here you can address, so you can, within the autoloader, address different grid types. So having 0.1 or 0.3 and so forth for tomography, for negative stain, SPA, and so all the recipes are set. Okay, now a little bit more to the material, reference materials. So since the launch of the system, those are just like last 2 years of publications, probably not all. I just selected, highlighted a few of them, which were nice. As shown in the previous talk, so there's a quite steep increase in, in the publication of cryo data, also on the CryoEM side. And we're currently holding the world record with the boring apoferritin. It's at 1.1 with a non-corrected, non-monochromated system, just a standard off-the-shelf cryo arm. It's quite nice. Well, it's apoferritin. Also looking at a bit more complex structures like beta-gal or laxegene, or whatever you want to call it. Also JOL with 200 and 300 kV systems are there with the highest resolutions. So quite nice, at least to benchmark the systems. Okay, here are just some structures. So I did some structures around the 2 angstrom area from 300 and 200 kV systems. This has been done in Glasgow, one of the very early installations here in the factory. Then a small protein 2 years ago, it's hemoglobin, 64 kilodalton, with a very high speed already at this time, like photosystem 1. Also tomography, I didn't mention yet. It can be done with the systems. This has been shown here, but as mentioned in the previous talk, you need to do some FIB milling, especially if you want to do subtomogram reconstruction or averaging. And paste tomo has been developed with on our systems at Tokyo University, and this is quite nice to orient, like to get an idea of where your lamella, how your lamella is oriented in the microscope. So it gets like a matrix, and then you can define different positions on your lamella and directly acquire tilt series on those different positions in parallel. Okay, yeah. Well, what is the workflow with GOL in terms of dual beam systems, FIB systems? So we have 2 sets. This is a very straightforward, easy set. So we have the JIP 4000 plus. You just directly take your site-engineered cryo holder into the FIB and prepare your lamella. So once you clip your grid, you never have to deal with it like unclip, changing it anymore. You just go to the microscope. That's quite nice. It has a very handy small chamber. It's price-wise pretty attractive, so that's a very straightforward solution. So on the cryoholder lamellar preparation. And then there's a bit more like steps involved. If you want to go with correlative work, we use an icon and a Lincoln stage here. We deal with the cryo-arm cartridges from blanch freezing and all the Leica components through the FIB and then to the cryo-arm. So you have the choice how you want to work. Okay. Also here, some nice publications have been done with respect to like the pharmaceutical applications. So, and using AI-based algorithms to detect like binding partners. This was quite a nice study done on the cryo-arm. And also a very high resolution structure of the FDS set. So the division ring for bacteria has been released recently, done on the cryo-arm. So yeah, okay. We mentioned micro-ED, and micro-ED is, or microcrystal electron diffraction is quite nice, as shown by Marta. And here I just want to show the benefit of the energy filter. So this is a system without energy filter, and this is one with. So, and you see, especially at the lower frequencies, you get much more information with the energy filter. So quite useful. Okay, I think, yeah, that's the software we're going to use for micro-ED, a GL-owned software. That's straightforward to use. Okay, here are some application examples for 200 kV systems. Cryo-arms have been released, so like ribosome, beta-galactosidase, and again, ribosome structures from 200 kV. I mentioned in my title a bit more an outlook. So we offer with our systems now electron dose modulators. So you know beam blankers normally are rather slow and they smear. Here we have the dose modulator which allows ultra-fast shuttering. So the setup is like this. You have your shutter, and this one is electrostatic. And then you have a dose modulator, and with this one you can, indifferent of your alignment of your TEM, tune the dose. So that's quite nice. So if you look at the movie on the right-hand side, that's a beam on the screen, and here's a knob, and with this one you can dim, it's like a dimmer in a room. Yeah? You can just dim and increase your brightness without touching any alignment. So it means you start your recipe for data collection, set up your beam, everything, and do not worry at all about the dose your camera needs or your sample. This you do at the end by simply turning a knob. So it gives you quite some freedom, and we allow to configure this dose modulation depending on your sample. So if you have thicker areas, you can apply different dose compared to thinner areas. So this is quite nice and can be ordered with our systems. Okay, so with this I'm at the end, and yeah, I hope I showed you some of the key features of the CryoEM. If you have some more questions, don't hesitate to ask, we are around. So Yusuf is sitting here. Okay, thank you for your attention.
Thank you. In the interest of time, we will have time just for one question, if there is. Yes.
I didn't know that you could reload as you go along, so that was really interesting. But how long can you run the instrument? Can you run it 24/7?
Yeah, of course, definitely. Yeah, yeah. It's fully automated in terms of liquid nitrogen supply. So yeah, it gets delivered with a 150-liter liquid nitrogen tank. So it runs around 1.5 weeks, and then we have to refill the tank. So yeah. Mm-hmm.
Okay. Thank you so much one more time. And we are finishing with one last sponsor talk. Here is Joao, and he's going to tell us about mass photometry, which is really not cryo-electron microscopy, but can give us some good clues about the quality of our sample, which, as we know, is important for any kind of imaging or high-resolution data collection. So, the floor is yours.
Does it work? Yeah. Yeah, I was going to start by saying, and now for something complementarily different, because it's really not CryoEM, but hopefully it's a perfect tool. Aha.
Okay. Let's see.
It's not the computer, at least.
Yeah, it's because it's missing the...
Aha. So my name is João, and that way, while we wait for this to work, I'm the technical sales specialist at Refine, and Refine is a company based in Oxford that was founded in 2018, and it developed a technique called mass photometry, which is again really not cryo-EM, but is hopefully the perfect tool to screen the quality of your samples before cryo-EM, as I hope I can show you. So what is mass photometry? Like the name implies, is a technique that uses light to measure mass, or in this case, the molecular mass of samples in your solution. And it really delivers a very rapid, accurate mass measurement without you needing to label any of your samples. And it will measure it single molecules in solution and in their native state. So it's really the perfect complementation for structural techniques. And because you have all of these nice keywords and buzzwords, it actually gives you access to subpopulations, which is not usually accessible with other techniques that can give you complex stoichiometries or oligomeric states of stuff in your solution. You can monitor complex multi-step processes. You can detect low-abundant species, or just use it to characterize purity or heterogeneity of your sample. Whether you do want to use it for sample characterization, interaction studies, or analysis, you can use it for most biomolecules, and these are all biomolecules that we've had the pleasure to see in publications. So within our detection range between 30 kilodalton and 5 megadalton, you can go from proteins, big protein complexes, bigger macromolecular assemblies, to up to nanostructures and even small viruses, or nucleic acids, as long as their molecular mass fits within the range that we can detect at the moment. So how does this work? Mass photometry is actually an interferometry scattering microscopy technique. And so it uses light. So you have a glass surface, usually a cover slip, that is illuminated by a laser. So you pipette your solution on top, 20 microliters, not more than that. And then as your biomolecules adsorb to the surface or interact with the surface, they will scatter light. And this scattering will interact with the light that is being introduced by the laser. And this creates this point spread function, or these dots. So each of these dots corresponds to a single molecule landing on the glass slide. And you can see the dots have different contrasts, and the contrasts correlate directly. So they scale linearly with the mass of each of the molecules. So if you have a calibration curve, you can pretty much have a single molecule detection mass scanner. Let's put it like that. And so the system just records each of these single landing events over a minute. So it's a video. And then if you have a calibration curve, it will just give you a distribution of the mass of everything you have in your sample. I actually have a video because I mentioned it was rapid, and I mean really rapid. Each sample takes, let's see if the video works. It does. Each sample takes roughly 5 minutes from this step to actually getting the histogram. So you just pipette your buffer to hydrate the surface, you mix in your sample, and you can see we're talking about 10 microliters of sample and 10 microliters of buffer. You close the lid, the laser goes on, and the video is recorded, and you can see the single landing events there, like in the illustration before. So it's really, really quick, very easy. This is the whole procedure. We have a running joke in the company that our CFO can run this, and he actually can. It's really just, if you know how to pipette, pretty much you can run the instrument. Because we're talking about mass, which is very universal, it's very easy to also analyze. So it's very intuitive data analysis. You're just looking at distributions of mass. It takes very low consumption of, takes very low sample. It's very low volume. It's also very low concentration. Where the sweet spot to get this beautiful single landing events is around 10 nanomolar, and it doesn't take a lot of space. It's a benchtop instrument, so it doesn't even take the full bench. Yeah, so there are many applications. I have like 80 slides. I'm not going to go through all of them, obviously. I chose 4. I hope they will be interested. But just to give you an idea of the type of things you can do, you can look at protein aggregation. If you're looking at viruses, you can look at fill ratios, empty full ratios. Just look at sample purity, binding affinities, assembly mechanisms, complex stoichiometry or composition, if you have different things mixed in solution, or just oligomeric states of your proteins or biomolecules. So the first example I have, and this is my favorite slide, is really, this is what this just everything it is this. So this is just an antibody, it's an anti-HIV antibody. And in this case, we just wanted to see if it oligomerized or not. And you can clearly see, just a Gaussian fit to the histogram with the monomer at 150 kilodalton, which is what it's described to be. Then the dimer, then the trimer. And then you can see the reason why we put this lower part is to show that the subpopulations are really easily accessible, because a single molecule, if you see a dot there, it's because there is a dot there. There's no false negatives. There's not enough number to fit a Gaussian, but if you would zoom in, you would see the histogram, very low number, show up. So it really gives access to very low abundance species. Likewise, if you want to see interaction between species, this is of course the simplest example, but you can do it much more complicated things. Just antibody antigen binding. In this case, it's Herceptin and its antigen HER2. You can see the in this case the diagram, but of the distribution of HER2 and then the distribution of the antibody. This is a great example because you can see the antibody, even though it's very pure, you can still detect probably just heavy chains that are still in the solution, and it's something to keep a look, to keep an eye on. And then when you mix them, you can if you mix in different proportions, you get different things. So if you look at the orange, which is the 1-to-2 binding, you see you have an excess of the antigen, and then you have some mixtures there. So you can see the purity is not perfect, that you could see also there. But then you see a peak at the 1-to-1 binding and another peak at the 1-to-2 binding, and because we're just looking at mass, it's just a matter of adding masses and seeing if it fits, if it's in the right position or not. If you, on the other hand, mix it on 1-to-1, then of course you will have an excess of antibody, and then the predominant binding modality is 1-to-1, and you can see the peak showing up. You can use it just to optimize purification processes, as simple as that. This is an example that comes from the Weizmann Institute. They were purifying 20S proteasome, which is around 700 kilodalton. So you can see here in the gray area. And it's a multi-step process. So you have gel filtration. You can see the initial gel filtration doesn't really have much. And then at the late elution, you can see a peak starting to show up. Then you go for a second chromatography, probably. In this case, more specific. So you start seeing an enrichment of your sample, and some of the impurities still there. Then you do a buffer exchange, or they did a buffer exchange to clear up some of the material that were not the 20S proteasome. And in their case, they were actually doing a last column, which they found out was actually detrimental to their purification process, because we can see here the impurity started going up again. So they stopped doing this fifth step because they got up to 75% purity with this process. And this is something you can easily calculate. I remind you that each of these steps took 5 minutes to do. So much more complicated, and this is an example more related to the CryoEM community. So this is the purification of the anaphase promoting complex with cyclosomes. It's again a purification step. And you can see this is a 19 protein complex, so it's not as trivial as just a 1-to-1 binding. And there are many subforms or incomplete forms that can be found, because it's sort of always measuring the full range from 30 kilodalton to 5 megadalton. You can see always the distribution, and again, it's a 3-step purification process, and the affinity 1 gives you a distribution. Then of course they pick up the fraction that has the full complex they wanted. They do ion exchange, and again you can see the enrichment of the full complex there, and you can even see that these actually form dimers. And the way to compare this, so usually this process is done by TEM, so each of these would take a few hours at least to go through. So it's negative staining EM, but in our case it took 5 minutes instead of 3 hours for all of the fractions. But you can clearly see there is perfect agreement between what was seen in the gold standard, which is the negative stain EM normally, and what we saw with the mass photometry. So much quicker and lower sample and faster. Yeah. So all of the examples, or the 4 examples I showed, they were done with the 2MP, which is our broad range instrument. It allows you to measure the range that I mentioned already, concentration range usually between 100 picomolar, 100 nanomolar. Sensitivity is below 1 nanogram. Resolution does not scale linearly, so you can. It depends, of course. You need to be able to distinguish the 2 Gaussians, and those peaks those histograms don't fuse together. So at 66 kilodalton, you can detect if it's 25 kilodalton separation. At 660, you have, of course, a little bit more freedom. Usually, we're working with bigger things, so 60 kilodalton with a mass precision of 2% usually, and error at 5% for single measurements, which is why you do more. The whole family of instruments, so the 2MP I just presented, the SAMUX is a version we did exactly optimized just for AAV characterization, so that full empty ratio I mentioned before. And there are also automated versions so that you don't have to be there and pipette every single sample. This allows for 24 samples at a time in 90 minutes without you needing to be there. So it's a pipetting robot on top of the instrument, pretty much. And of course, we sell all the consumables, which are not that many. It's pretty much glass slides, ultra clean glass slides, because of course we're talking about single molecule. They need to be very, very clean for this to work, and then the cassettes so we can separate the samples what you put in there. So the running costs are very, very low. We're talking about €1 per measurement, more or less. Like I mentioned, we're a spin-off from Oxford. We now have a global presence. We have over 200 instruments around the world. And we really just hope our instrumentation can really help streamline all these complicated processes so you guys can bring the right samples to the complicated and time-busy techniques such as CryoEM. So I think it's a perfect partner for that. So thank you. If you have any questions.
Thank you, Joao. We have a question.
Thank you. I wonder how detrimental is aggregation. For example, you had the example with the antibody with the 2 binding sites. So if you would then link several molecules, you couldn't think of getting aggregation.
Yep.
But you say you have a range up to 5 million Dalton.
Yep.
So can you comment on that?
So I mean, as long as it fits, it will show up. So of course there we were showing just where we saw the hits, but if you would have had aggregation in that, you would have also something further down the line. So if as long as it fits, of course we can promise the linearity up to 5 megadalton. It will still measure bigger things, but at that point it will start being unclear for us if it's the mass measurement is correct. No, it wouldn't hinder. No, unless it's really massive aggregation, and we're talking about micrometer aggregates, but yeah.
Okay, I think in interest of time, we will have to stop here, but Joao is going to be here in front, so you can go and ask him whatever you want. And now we are going to have a break for lunch. There's going to be sandwiches served outside. And then we are going to start here at 1, as was planned. For the scientific part of the mini-symposium. So thank you all for now.
And I'm going to do cryo-electron microscopy. So Tom is goes, I don't know him. You probably know him.
He's busy and sometimes answering emails, which is frustrating for me.
okay. Good. And then I write the application down before. Do you have any tips, any goals, no, it's a free pro. I'm applying for free pro.
We actually, do you know what, do you know the
Okay.
So we actually just last month. So it's for
Hello, I want to introduce myself. I'm just a new guy here.
I think you want to
Yeah, initially.
Yeah, exactly.
So I just cannot say that I'm completely with
I will come in a second.
But he.
Okay. Yeah, we kind of, we can, because now I feel keen on it.
Yeah.
I think I pursue that as
Okay, thank you.
Did not go so well.
I'm not going to bother because I don't know if I know. I know what they are.
So the platform which is based on scripts, nicely organized at least these things I do.
Yeah.
Don't have to do like combine like writing.
So this is actually,
Yeah.
well, so I have the towers, and so I actually, so we had been running FEI software on the towers, but then I installed CryoEM just now, and it's been quite cool actually to see.
Sure, it's cool. I like it actually, but you need to know what you're doing. You got some experience. And if you are running a larger facility and have like users coming back and forth with their samples, not having the time like to sit days to learn it and so forth.
So you can just install this on top and then...
Exactly. And it's giving you a graphical interface to do all the single particle data collection without like tedious CryoEM script starting and so forth.
Yeah, I hope you can
I think this was with the jails, so, well, of course, we all know there's kind of double fishes all around everywhere too. Well, with the cartridges and especially this kind of like switching from one, let's say from tomography to single particle, that's really cool actually, because that's something. I actually started on the JL100B.
Oh, okay. Wow.
Yeah. So, which is that's a while ago.
Okay.
That was I thought, and I still think that was just such a cool instrument. It was
used to offset some
you have a no, I have a 1230.
Well, it's an early version. Okay.
No, I think I have the old version of it, the old one. Well, anyhow, it's a
But is it
robust instrument. It's amazing.
That's the
day-to-day stuff, of course, we do lots of simple things too. So
good to see you. And where are you based? In Germany, same place, right?
Yeah, so
we met before.
Yeah.
2 weeks ago.
Oh yeah, the CryoET also, but even before that.
So I personally live in Hamburg, but our office, our demo center is in northern Germany. But I work from home actually. Because I'm responsible for Denmark, Norway, Sweden. So Hamburg is nicely in the middle between. It's a bit easier. In Denmark I can drive by car, which is quite nice, and then here it's a short ride. So.
But you're in the Munich review.
As application team, so we are around.
Yeah, that's right.
Okay. Good.
Yeah.
Yeah, actually, so,
saw Yoshi's microscope in Kyoto.
Okay, cool. Yeah, that's
big, and it was actually
Yeah.
that was the top of the line microscope.
Yeah, I don't
Yeah,
know.
the inauguration of the TM from us. Who was there?
Inauguration.
In Delft, no?
In Delft.
Okay, I met
Oh, he was there just for the
just for the
inauguration.
inauguration.
Was
Yeah,
from
giving a
Fania?
talk.
The Motorarm?
No, 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 Fujiyoshi, Mr. Yoshi, a user in Japan.
Okay.
So we're listening. Oh, yeah, at the conference.
Yeah, he was really a pioneer
Yes,
in
yes.
cryo.
And he's still actively developing products. So
Okay,
he's
and he's in
still-
Nagoya now,
yes,
right?
yes.
Yeah. Okay,
And
so he's still there.
Yeah,
If
still
you-
on.
if
Still
you-
on, yeah.
If you just say hello to him.
Yeah, okay.
I will also.
tvilsomt forum.
Well, it's not for now. So, did you try to check with the people at the reception? So it's not for this event.
Men det vel sånn. Snakker du norsk? Snakker du norsk?
Nei, svenska.
Jeg skal levere til noe som heter Forum møter om. Her står jeg. Vil du si det ikke her? Da leverer jeg det her. Til oss, eller?
It's not for us,
no.
is it?
Hvem det som skal statens pensjon? Det står her står det et mobilnummer, kursansvarlig Per Ivar Sprete.
Her den adressen.
Jo, men her står det Statens pensjonskasse. So det en helt annen organisasjon. It's a Norwegian State Pension organization who has a meeting here. And there's a mobile number here, so just call that number.
Yeah, absolutely.
Så bare ringer det nummeret. Jeg skal ringe det for deg, eller?
Jeg skal levere her, for det skal jeg lese noe litt.
Ja, det kan du si.
I guess...
Kom hit og hente da.
Ja, men det står 19. oktober. Kurs for 19. oktober. Det er 18, ikke sant? So they have a course here tomorrow because it says course 19 October.
Yeah, exactly.
So I can call that guy and just say that du kan sikkert bare sette igjen her, altså jeg vil tro det. I think you can just say you can just put them there probably, and I can call that. I can just put that number.
Det gikk fremover her nå.
Tusen takk. Yeah, absolutely, absolutely. We're all in line to do that, so one by one.
So Nicolina, you're probably busy. You want me just to send some text to this guy, say that there's a delivery for him?
Okay.
I can do that if you wish.
Thank you so much if you do that. I think it's, I don't...
It's probably some course materials. Like
yeah,
some,
nothing is going to happen. Nothing is going to happen to this, and it was not like the guy was not opposed because I'm thinking he could have technically.
Okay, this works.
I'm going to tell to the Forskningsparken person that this is here, so they just like can store it until tomorrow in one of these things. That's how they usually do that. So he's going to come here eventually. So I will tell him. So if you
I don't have to do that actually.
You don't have to do anything. I will talk with Forskningsparken and explain them that it's
probably it's not shrimp or anything.
Yeah. Yeah.
So he was a day early.
Yeah. I was like confused when I saw it. I'm like, are we expecting something else here? Yeah.
But it was the wrong date apparently.
Keyboard setup. You didn't write up the shift key. Have you done this thing before with the setup? On the Mac? I don't know. I can just try it, see what happens.
Yeah.
You can just cancel that.
Yeah, I guess so. And if you go in the presentation mode, it should pick it up. Okay, perfect, yeah. If you have some movies, maybe you want to try. Oh, you know what? They have actually these things here. Look at this.
You're in it?
Yes. No, but it's that I have watched it from the others. I think maybe I can. I should really. Okay, I can leave it for you. Doesn't.
And then you're all set. Yeah, just checking out the connection and then the buttons. Okay, great. Good.
You're the first.
Am I first?
I have no idea.
I didn't steal it from you.
Who's up first?
I think it's me.
cutting all ends, but it's very nice to please have this session.
Yeah, yeah.
Yes. Yeah. So you give us one, because the whole thing is 35 minutes, right?
Yeah,
Including discussion. So you get up in 25 or something.
yeah, I think 30
No, but then, yeah, but I need to finish by 30, right? So otherwise there is no question.
there are lots of people.
Yeah. Yeah. Okay. Yeah. Just make sure I see. That's right. Push me away.
These
Medical practice is also very, among others, and I was just talking a lot at the beginning of the it's going to be recorded and just for people to be available. But it's actually really cool and I'm going to demonstrate that it's not really only just like these atomic particles. It's really looking into tissue and exploring the properties of the plastic and then something to keep in mind. Yeah, thank you. Okay, thank you all. Am I on? Or does this work? Yeah. Let's see now. Okay, now better? Yes. Okay, good. Thank you all for coming to this second part of the Minisymposium. And so we are going to have a little bit more science presented here. But before we go to the science, I actually just wanted to touch a little bit on history of CryoEM in Norway and actually plans that we might have. And one of the things that was lingering in the air for a while, but you all know that we are all very eager waiting for the life science building that is actually taking shape now, which is really exciting. And one of the things that was planned for this building was also CryoEM facility. And we've been here at University of Oslo from our side working a little bit towards, being ready for this if it happens, when it happens. And I have first a couple of introductory slides, but I'm not going to talk too much about that. It has been pointed out already that this is becoming a leading technique in solving protein structures of proteins, and it was awarded a Nobel Prize in 2017. And maybe something that is fresh in the minds from all of us was a fight against COVID. And here actually the full potential of the technique came into a place because lots of molecules that are involved in the virus infection were actually solved using this technique, and lots of treatments were inspired to be generated with the help of the data that was obtained with CryoEM. So it's definitely something that has a place in a future science in developing, fighting diseases of all different kinds. And we never know when is the next one coming after us. But anyway, in Nordics this was also covered, so I will just breeze through that. We had a beautiful presentation about SciLife Lab, and this is probably already outdated. You have more microscopes than what I have here. Denmark also has 2 nodes with high-end microscopes. Finland also has 2 screening microscopes, so we are kind of the only of these big Nordic countries that is still without a microscope. what were we doing here at UIO to raise the awareness and to generate the expertise is we had a number of invited lectures over the years, and among others, even a Nobel Prize winner was here visiting us a couple of years ago, Richard Henderson, that gave a beautiful talk. We also started training our students and postdocs that went to UMIA, Aarhus, or in US to get hands-on experience with the technique, because we don't have it here. And we are also organizing mini-symposiums, this one being one of them. Also, data processing workshop is taking place soon after this in cooperation with Aarhus University. And we are planning to have courses that would be also focused on cryo-EM, so that the people learn this technique now early on. How does it work in terms of doing cryo-EM at UIO? So far for sample freezing, we have VitroBot at IBV. We are doing our screening and data collection either in SciLife Lab as a part of the bag, as was already mentioned, or also people can apply through a different other competitive places to do it in other facilities. And so far, we were paying 5,000 SEK, and now the price increased, and they're squeezing us out a little bit. But anyway, it's still pretty affordable. That's not the biggest bottleneck. And then the other bottleneck that we have is a little bit of with data processing, that also we need stronger machines for this. So basically, in terms of funding, this was funded, VitroBot was funded by NCMM. For data collection, we get funding from core facilities funding, and this was our own funding for the data processing, and we are kind of reaching out the capacity here. So current bottlenecks are screening of the sample, so we can freeze, which is great, but we cannot screen them. We have to send it out and pay for the screening. Data collection, okay, and then data processing is also a bottleneck, so we will need to invest a little bit more in the data processing equipment. But for now, just, yeah, elevating these two, like screening and data processing, would already mean a lot for the community. In terms of the interest in Norway, so there are a couple of groups that have already collected the data and published data at UIO. There are also active groups at UIB that are actively collecting and publishing data, and there are lots of people that express their interest in both UIO and UIB for if the equipment would be available that would use it. And we are also in contact with our colleagues at UIT, NMBU, UIS, NTNU, Oslo-Met. There are people that are interested in techniques in all of these universities, and if there would be equipment, would be happy to, to use it. This is our vision, basically to have, to start off with 2 microscopes, 200 kVs. These are probably wrong pictures if Thermo Fisher people are here. But we would like several nodes to have VitroBot, and maybe more than 3. But 2 main nodes that would have the screening instruments of 200 kV, and that would be at Oslo and Bergen. And then we are also exploring this possibility, the same as in Sweden, of using the national computer resources at the national basis for the processing of the data. It needs a little bit of work, but it's not impossible. So this is where we are right now, and we are hoping that we will be funded soon. Okay, in terms of like particular work in our own lab, I'm just going to quickly show a couple of slides. Everything was done by Ahmad, who is here in the audience, with a little bit less hair than on the picture, but a lot more knowledge. So he's our expert. We are working on centromeres, which are parts of the chromosomes that are important for cell division, because that's where the kinetochores formed and attach chromosomes to separate them in different cells. For the organization of this loci on the chromosome is important one particular histone, which is a variant of the histone H3, which is present in nucleosome of all our chromosomes, but at the centromere we have the presence of this particular histone. And we were trying to use CryoEM to see what is so special about this histone, what kind of special properties it gives to the nucleosome. And so this is a CryoEM map from our paper, where CryoEM map of regular nucleosome and nucleosome that has this special histone variant. And you can see the red parts are low resolution, meaning more disorder. So we have basically shown that this nucleosome has more disordered DNA ends, and then binding other centromeric proteins, these DNA ends get even more disordered. And based on this, we proposed that it would array of this type of nucleosomes would infer a different chromatin structure, which would be permissive for forming of the centromere and kinetochore, which is the big complex that is necessary for attachment to the microtubules. And recently, we have another paper that was all collected data was all processed by us and collected by us in SciLife lab. And here we have also obtained the cryo-EM maps of the nucleosome in complex with yet another protein called SAMP-B. And you can see that in comparing to the regular nucleosome where DNA ends are closed, SAMP-A nucleosome where DNA ends are more open, and then in the presence of this special protein, SAMP-B, we actually see several classes of proteins, some of them with a very open DNA, saying that presence of this special histone plus another protein kind of really generates this open chromatin that is necessary for the formation of the complex. And with that, I'm just going to finish and thank all people in my lab, and especially Ahmad, who is really driver of the process and expert in cryo-EM by now, and also people in SciLife lab. We collected most of our data in Umeå, and Linda and Michael were really instrumental there. And that's all from me for now. Thank you. Okay, and the next on the agenda is Matteo. And Matteo is going to tell us about PX Oslo, which is a structural biology core facility here in Oslo. And, oh, it's connected.
Just the ports on the side.
Okay. Okay, good. And, yeah. I forgot to say, are there any questions?
So yeah, as Nicolina said, I'm Matteo. I'm a PhD student working with Ute Krengel, and I'm the ambassador for the UAO structural biology core facilities. And I think I also will have a bit of time in the end to show you a bit of my own research with cryo-EM. But first, let's focus on the core facilities. So the core facilities are formed by 5 different groups here at UAO, involving the Krengel group, working with carbohydrate interacting proteins, mostly with protein crystallography, the Hesleth group that works with metalloproteins, redox-active enzymes, and has expertise in spectroscopy as well as protein crystallography. Nicolina's group, about whose work you've just heard a bit, and has expertise in cryo-EM mostly. Then we have the Lund group that works with soft matter, mostly with scattering methods, small angle, X-ray scattering, and also neutron scattering. And then the Dalhus group at the hospital, which works with DNA protein complexes and different proteins involved in DNA repair. So from this expertise is combined among different places at the university. So we have the Department of Chemistry at the University of Oslo, also the Department of Biosciences, the Norwegian Center for Molecular Medicine here at Fornix-Parken, and also at the Oslo University Hospital, as I just told you. We offer a bunch of different services related to structural biology, all the way from construct design to structure determination. This works with the work starts with protein production and purification, and then of course afterwards crystallization and X-ray data collection if we're using crystallography, which is our main expertise. But not only, we have access to international facilities involving synchrotrons, but also, as Nicolina just pointed out, for cryo-EM. And we have a bunch of different tools to study, general biophysical tools to study proteins and protein-ligand interactions. And we can also provide advice in other techniques such as NMR or small angle scattering. We can also provide expertise with diffraction data processing and modeling, general protein spectroscopy, and we organize courses in structural biology and protein crystallography at UAO. In our facilities, we have plenty of tools for protein expression, purification, and crystallization, including ACTA FPLC systems for purifications and different liquid handlers like thermoelectric liquid handler, crystallization robots, Oryx and Mosquito, and crystallization plate hotels that facilitate following the growth of crystals on a larger scale. And then we have a bunch of different machines that, as I told you, allow us for biophysical characterization of proteins, and most of them are at Oslo University Hospital, and allow us to do techniques such as MST, ITC, DLS, NanoDSF, SPR, and SEG-mols. And then also at NCMM we have hydrogen deuterium exchange MS possibility for that. You can find more information on our website. And also, you can always ask me or any of the responsible professors. And that's all regarding the core facilities. And I'm going to show you a bit of my own research regarding cryo-EM for my PhD. And this is basically a collaboration in which our collaborators from NMBU and University of Bergen have been working with this protein for a long time, and they come to us trying to see if we were interested in solving the structure of this protein. And the technique of choice was cryo-EM, and for that we went to Seiler's lab in Stockholm, about which of course you've already heard a lot today. And then together with some other collaborators from Germany, the University of Osnabrück, we managed to get a final reconstruction. So the protein that we were interested on is vitellogenin, which is this protein from the honeybee. It's present in all egg-laying animals, and in the honeybee, it's a very important protein that has many different functions, and it's the main circulating protein in the hemolymph, which is the blood of the bee. So it's the main precursor of the egg yolk, but other than that, has many other functions that are related to metabolism, also social behavior. So the levels of the circulating protein determine if the bees are going to go foraging or going to stay in the hive and have different social functions. It has also been shown that it's involved in aging and how fast bees die. And it's shown to be protective against oxidative stress. And finally, but also importantly, vitellogenin has developed a bunch of immune functions that are very varied and very important as well, and seem to be very important And it's why we find this protein not only in the females that are laying eggs, but also in sterile animals, such as the worker bees or the males. So this is the structure that we managed to get. Interestingly, we got the protein directly from the hemolymph of the bees. So there was no recombinant expression, which allows us to see more stuff that we have produced it recombinantly, and also it's a very complex protein and it's very difficult to, or like our collaborators haven't managed to produce it recombinantly. So we managed to get this reconstruction at 3.2 angstroms, and you can see it here colored by domains. And down here you can see the different domains, and also the parts that we can actually see in our structure are the ones that are colored in the lower representation. So we're basically missing some flexible loops, and importantly this domain in the end, the CTCK domain in purple, we're not observing it because it's connected by a very flexible loop, and it's not really attaching anywhere in our protein. But basically we have this big lipoprotein. We all can observe this big lipid binding cavity here that is formed by these beta sheets around that are formed around, and then also this extra domain that was never solved for a protein of the family of vitellogenin, which is very interesting. I'm going to tell you a bit more about it later. Here are some details about the structure. So we can see some lipids around the lipid binding cavity, or densities that probably correspond to lipids, and a phospholipid head. A very nice glycosylation, which we could observe because we obtained the protein from its native source. And then also some zinc binding sites, which is a protein that binds a lot of zinc, and this is something that was known before, but here we could see a bit more of the structural basis for that. We also very interestingly observed the cleavage product in the same amounts as the bigger particle, and we still not know, we're not sure about the biological significance of this. It could be just something that has happened post during the purification. It's not really relevant, but it could be very relevant because vitellogenins are known to have cleavage products that have functionalities, and fragments of this size have been described for the honeybee to have biological relevance. And finally, I want to talk about this von Willebrand factor domain that we described for the first time, and also the CTCK domain, which we could obtain a prediction for how it folds using AlphaFold. And these two domains are found in other proteins, namely the von Willebrand factor, which is found in the blood, and also mucins in the intestine. And in these proteins, these two domains, what basically produces these long concatenamers of protein that have very specific functions, like physical functions. And basically, the CTCK domains aggregate dimerizes with the CTCK domain, and the von Willebrand factor D domain dimerizes with itself through these disulfide bonds. The interesting thing here, however, is that vitellogenin, even though it has these domains, it doesn't have these disulfide bonds that are involved in intermolecular interactions. So it's really puzzling why vitellogenin would have these two domains, but not the main structural elements that these domains are known for. So, as it usually happens, solving the structure has led us to ask more questions that need further studying, but so far we haven't had time for more. So I just need to thank all the people that has involved with you and you for your attention. Thanks so much. Yeah. Thanks for freezing the grids, by the way. Yeah.
Oh, I forgot what I wanted to
Yeah, it's a long time ago.
Yes, I was thinking, so you have, why is it, okay, one stupid question, why is it called vitellogenin? If it's in bees?
In bees. Like,
Yeah.
I think vitello in Latin or Greek is the yolk of the egg.
I see, yeah.
So,
that's where they come.
Yeah.
And then,
you can also ask me later if it comes back.
Exactly. Maybe if I see the slides.
Yeah.
Too late, sorry.
Yeah. Okay, so let's thank Matteo one more time. And our next speaker is Flora. And Flora is going to say a little bit about BioCat. BioCat is also one of the sponsors, and maybe I'm going to, while she is setting her computer up, I'm just going to actually use this opportunity to thank all of our sponsors. Thermo Fisher and Geol and Refin, and also PX Oslo and BioCat. And BioCat, you will hear more about what BioCat is during Flore's talk. So, floor is yours.
Yes. I don't know. It's probably turned off. No, it's on.
It's on?
I don't.
Maybe it's on the wrong side, I think.
It's supposed to be like that.
Yeah. We can hear your hair, so I think.
I hope you can hear me. Otherwise, I can shout. So, as Nicolina just said, BioCat was one of the sponsors for this event. So, what we are is a research school that focuses for PhD students. It was funded by the Norwegian Research Council. And what we want is to provide the best experience for all the PhD students within Norway. So give them some experience in the industry by having industry talks where they present what they do, how they do recruitments, how to apply to a job in the industry. We also offer a lot of courses in basically bioscience, so it can go from everything from protein crystallography to enzymology. We also do a lot of transferable skills development with courses in science writing, in scientific illustrations, and we invite usually very many great people from abroad to give those courses. We give a lot of networking opportunities. We have an annual conference every year where we try to all meet together and present what we do in our research. And we also give some travel grants, which is one of the reasons why BioCat is so popular in Norway. So, as I just said, we have had already many presentations, and more than this, we try to have this very often. There was a bit of a lagging time during the summer because it's difficult to get people to be present during the summer, but we will start up that very soon. And as I just said, we fund travel grants and research visits. So if you have to go to a conference somewhere, you can actually apply for funding, and they will help you fund the travels there, like the visa fees and so on and so forth, but also research visits, and I will come back to that when I will present a bit of my work, because I benefited from that myself. So just a tiny bit about why CryoEM is great and how we actually managed to use this amazing, I would say, Nordic collaboration power to be able to get the CryoEM structure of this new chimerolectin. So our protein is called CCTX2, and it's found in the vegetative mycelium of the protein. And when it's challenged by a nematode, the protein will be, of course, ingested with the rest of the cells and will enter the epithelial cells of the nematode. And upon entry, it will distract the complete stomach tract, intestinal tract, but we still have no clue how it works and what it resembles. So that was one of the questions we were addressing with our collaboration partners. So the protein is made of 5 different domains. It's about 90 kilodalton. What we know is that the 2 first domains are of lectin function. So it means that they are capable of binding carbohydrates, and that's the way they use to enter the cell. And they bind what we call the glycocalyx. So this is this whole sugar network that you have at the surface of every cell. And in our case, they recognize very specifically this lactinac component, so composed of a galactosacetamide and glucosacetamide molecule. The 2 next domains, we actually have no clue what they do, but we know that they are supposed to harbor the same ricin B chain-like fold, which is formed of this beta threefold symmetry. And the 5th domain is of completely unknown function and unknown structure. So that was a lot of questions to answer. Luckily, with Gabriele sitting just there, and of course with Ahmad's help, as I said, collaboration within the Nordics, we managed to solve the structure to 3.1 angstrom. And what we found is that we have the 4 first domains that are basically serving as a base, and then we have the 5th domain that sits on top. And from there, when we look more, we see that we have, don't have a pointer. So we have our four domains, and the two first domains, we even see some residual density that could actually show the galactose molecules that we use to purify the proteins. So there we might have some ideas of where the actual lectin binding sites are. We have also in magenta the linkers between the different domains, and then we have the fifth domain just up there. And here we can also see for all the different domains, so it's always an overlay of 1, 2, 1, 3, and 1, 4 of this very nice beta threefold ricin B chain-like fold. So what we assume is that the two last domains might have been ricin binding domains once upon a time, but with mutation and substitutions of amino acid, they actually lost the capability of binding, but still are there for holding together the whole protein. And then when we look a bit more into the fifth domain, so we see that we have these beta sheets at the these beta sheets here that is very central with the two helices. And otherwise we have different subdomains with very long linkers. And here in red there is one very important thing, which is a caxin cleavage site, which is a protease that you can usually find in the trans-Golgi apparatus. And then we have here in magenta an ER retention sequence. So that means that the protein eventually could travel all the way to the ER. But since it's buried in the protein structure, it's most likely not available like this. So one of our hypotheses is that the cleavage for caxin is necessary to basically free the fifth domain, expose this ER retention sequence, and that in that way the protein would be able to travel all the way to the ER and then get in the cytosol, get refolded the way cholera toxin does, that we don't know yet. But it seems to be necessary because we have functional studies that shows that if you mutate these parts, then the protein loses toxicity. And then I just said, very important, the collaboration. So during my PhD, I went to Denmark for 2 months, tried to improve the structure that Ahmad and Gabriele already had built. I collected some very, very nice 2D classes, and I didn't manage to improve the resolution. So sometimes failure is still a learning process, but it would be very helpful to be able to do all these screenings and great preparation on site in comparison to traveling all the way to Umeå or somewhere else to Denmark, and before being actually able to see whether it would be applicable in your project. So thank you to all the people who have been involved in the project, and if you have any questions about BioCat or that.
Thank you very much for a very nice presentation, Flo, and also Matteo before. And I want to again acknowledge BioCat's funding, and we forgot one funder, so I should like to mention that one here. That's Hydro's fund, because they gave quite a lot of money, so we are very grateful for that. Now, some of you might know me. I'm the leader of the structural biology core facilities, and this whole event here and the explosion of CryoEM reminds me a little bit of when I started crystallography in '88 for my PhD, because hardly anyone was doing this. And now there's every better university has at least one crystallographer, and that's what we are experiencing now with CryoEM also. And it's my pleasure to introduce the next speaker, Gunnar von Heine. We heard a little bit about how you got into CryoEM, so from a very unexpected side. And I read a little bit about your CV. What I didn't know before, for example, that you actually had a PhD in theoretical physics. So this shows that people who come from completely different fields can become convinced users and applicants of CryoEM. And then you have been being director of bioinformatics, as you told us, Biomembrane Research Center, and you built up CryoEM in Sweden with SciLife Lab. What another unexpected thing from your CV was that you were actually science correspondent for the Swedish National Radio
Long time ago.
long time ago. So, and I know that you're a very good speaker because I heard you once before when I was in Gothenburg. That's when I learned about the positive inside rule about how proteins have the topology in membranes. And you have a long list of awards. I'm not going into that now, but once in a while I have seen your name on the invitations to nominate for a Nobel Prize in chemistry. So we see you are doing a lot of great stuff for the community. The floor is yours. Thank you.
Okay, I hope my mic seems to be working. Thank you. So what I thought I would do is I'll tell you a bit about work we're doing right now and have done for the last 5, 6, 7